Well, good morning. It's nice to see everybody in the room, and welcome to the second annual Animal Drug User Fee Educational Conference. This is a public meeting, and the information that will be presenting will be recorded and ultimately be in the docket, as well as the video will be available on YouTube. And I'll get to more of that later. But to get the meeting rolling, I would like to take this time to introduce Dr. Matt Lucia, the Director of the of new animal drug products evaluation. Matt, come forward.
All right. Good morning, everybody. As Walt mentioned, my name is Matt Lucia, and I am the director of the Office of New Animal Product Evaluation here at the Center for Veterinary Medicine. And I would like to welcome all of the attendees to the second annual ADUFA 5 Educational Conference, and would like to take a moment to acknowledge both the in-person and online participants. Thank you very much for joining us. We have approximately 320-ish members of the public registered to attend this year's conference, with about 35 members of the public joining us in person and about 285 members joining us online. So very nice to have you all. So today's conference is intended to build upon what we did last year, which was the last year's, for those of you who remember, last year's conference was basically a new animal drug approval 101, or the basics, a high-level overview of the drug approval process. And so we are looking this year to build upon some of the principles that were outlined last year, and believe these topics that you're going to be seeing presentations on today do directly impact the regulatory review and approval of new animal drugs. To that end, the 5 key areas that you're going to be hearing presentations on are going to be the ADUFA fees and waivers, foreign data, real-world data and evidence, high-quality submissions, and adaptive study designs. Just a quick note, the agenda item for the ADUFA fees and waivers actually was one that was generated and came from your all's questions last year. As part of the presentations last year, we received several questions regarding ADUFA fees, and so that was the progenitor of this session's, one of this session's topics specifically that came out of last year's meeting. So the topics selected for today's conference represent items that are areas of frequent discussion at CVM that we have with industry, and we believe that additional clarity and practical guidance can significantly help benefit the drug approval process in these areas. So the goal for today is to not only provide useful information, but also to include practical real-world examples of how these different subject areas have been used or can be used to benefit the animal drug development process. The presentations will also include potential challenges that can arise and provide some examples of products that have successfully navigated and worked through these challenges toward approval. As we did last year, we will be hosting Q&A panels after the presentations for both the morning and afternoon sessions, and we encourage you to actively participate by submitting your questions throughout the day. So Walt, well, as he mentioned, he will be providing some more logistical administrative information on how to go about submitting your questions using the new process that we've established this year, again, learning from our experiences last year. And while not every question will be addressed during the conference, unanswered questions will be captured for consideration in other venues or potentially for future agenda items, as again, we did with the ADUFA fees and waivers agenda item for today, which came directly from the questions we received at last year's conference. Before we get started with the morning session, I'd like to take a moment to offer some acknowledgments and gratitude as these conferences take an enormous enormous amount of work to put on and could not be done without the very dedicated and significant contributions of so many people. I would like to express tremendous gratitude to the logistics team, specifically Walt Ellenberg as the committee leader, Martine Hartagensis, Joe Faust, Elise Moreau, Jim Delaney, Lisa Troutman, Brandi Robinson, Jendai Robinson, Brian Weiss, Julie Putnam, Adam Stamper, and Cam Curie. I'd also like to acknowledge the collaborative efforts required from all the presenters, subject matter experts who provided technical guidance, and leadership that reviewed and finalized the presentations. Extraordinary effort goes into putting on these conferences, and they do a wonderful, wonderful job. And also thank you to all of the people who've handled the complex logistics required for a large hybrid meeting conference, including our IT colleagues who are helping to run today's conference. Without them, this would not be possible. So thank you so much. And I'd also like to recognize our industry colleagues who helped to identify valuable information areas and assisted in developing today's informative agenda for the conference. And with that, I'm going to hand the podium back to Walt, who will provide you all with some more administrative and logistic information for the conference. I'm looking forward to a great day of learning and discussion, and again, thank all of the attendees for joining us today, and from both in person and online for the participation in today's second annual ADUFA 5 Educational Conference. Thank you.
Matt's a good bit taller than I am, so I had to adjust. As I said earlier, thank you everybody for being here. I really appreciate it. And I really can't overstate the fact that this wouldn't be possible with a dedicated group of really, really good friends and colleagues. And so with that, before we get rolling into all the presentations, there's some stuff I need to really bring you up to speed on. As Matt alluded to, we've introduced some new information and new technology this year that should make the actual submission and receipt of questions much easier. And so with that, I'm going to go through a few points and lead into the Q&A question process. This meeting right now is being hosted on Microsoft Teams Town Hall platform. And for those of you who are online, the chat function and the cameras and the microphones for those online are all deactivated. So we won't be able to receive any information except for any questions that you receive through the Q&A process. My intent throughout this session is to make sure that we stay on time as best as possible. Some of the presentations may be a little bit shorter than is specified in the agenda. However, others may be a little longer. Our goal, though, is to make sure we stay online on time so that those online can actually follow along and know where we are in the process should they join at a different time throughout the day. For all those in the room, just a reminder, please sign in, and we would highly encourage the use of the name tags, just so we get to know who you are and you will know. We're all trying to wear ours, so it actually will foster the communication and collaboration and any kind of communications that happen outside during the breaks. Just so you're aware, the kiosk out in the main lobby should be open for food. During the morning, we'll have pastries and coffee and juice and water. They will provide lunch, cold cut sandwiches, wraps, chips, and drinks. And then in the afternoon, they will have beverages and maybe some sort of snack. All that is for point of purchase, and I believe it may be card only. But don't hold me to that, but that was what I was told. As Matt referenced, earlier, all of the questions that we receive during this morning's and this afternoon's presentations that are submitted by you will be captured and they will end up in the docket. Last year, we received over 100, I think the number was 108. We received 108 questions. We didn't have time to go through all the questions during the Q&A, but as mentioned last time, we honored our commitment and all the questions were held, maintained, put in the docket, and actually considered, as Matt said, during the agenda development process for this meeting. For those online and for those in the room, if you have a question with regards to the information on this particular meeting, like the final agenda and any kind of PowerPoint presentations or any other information that we have associated with today's meeting, they can be found at the link shown above at the CVM event page. All the information associated with this meeting, as well as last year's meeting, are in the same docket as was used last year. We plan to do the exact same thing for the meetings over the next 3 years. The goal in that is it allows us as part, this is all part of the ADUFA 5 goals letter that we have, that was put out during the negotiation, after the negotiations were concluded. It's much easier to keep the information associated with the requirements set forth in that letter if we keep it all in one docket, so it doesn't get spread around. So you should always be able to refer to this location, this docket. After the presentation, since it's being recorded, we will place it on YouTube. The actual recording will be indexed so that if you're looking for a particular topic that you want to review and see over again, however you want to do it, you'll be able to go to that section of the video on YouTube and it will pull the actual presentation right up. And so now here we're going to get into something really pretty new, at least new to us, and that is the use of the QR code for the Q&A format. Just so you're aware of how we're going to proceed, 30 minutes prior to our break for lunch, we will have a Q&A session, and then 30 minutes prior to the end of the day, we'll have another 30-minute Q&A session. During the day, however, everybody in this room, as well as anyone online, can submit questions pertaining to any topic you want to discuss associated with the meeting. We're using it by means of a QR code. Now, for those who are in the room, the QR codes are on the walls and on the tables. You should be able to access the QR code through your phone. If you can't access it readily inside of the room, you can still probably go outside and access it depending on your cell network's strength and signal. Once you get into the the QR code, it would actually take you to a page that I'll show you in just a minute. But I want to emphasize a couple of things. Just so you're aware, any questions or all questions that we get through the QR code process are anonymous. There's nothing that was going to indicate who you are, who you're associated with, everything. So it really breaks down any barriers that you may be worried about asking questions that you really don't want others to know about, but that happens. We will answer as many questions as we can during the morning Q&A session and the afternoon Q&A sessions. Again, we will retain all these questions in the docket, and they'll be used as part of the development of next year's agenda. At the end of the meeting, we will deactivate the QR code. So it's not like once you leave the room, you can continue to ask questions. We have to have a closure on that. This is the QR code. For those of you who are online, you should see 3 pods. One pod should show the presentation itself. One should show the video of the speaker. And then we have a just static image that shows the QR code. You should be able to take a picture of that, and it will take you to the following page. This is the page that you should see whenever you activate the QR code. All you would do is go into this field, you click on the topic of your question, and then it is a free form. You can type in your question and hit done. That submits the question. We then receive it, and we can then file it so that I can use those questions during the Q&A process. You are not limited on the number of questions that you can ask. Once you hit done and go through that process, it actually will give you the option to ask another question. When it does that, it will take you right back to the page that you see here, and you can then ask other questions throughout the day. So again, you're not limited. So today's agenda, there are copies in the room, but I'm providing this online just in case folks haven't seen it, is fairly straightforward. As soon as I'm finished my presentation, we'll introduce Aila Albrecht, who will come up here and talk about overview of user fees and waivers. Then we'll move into foreign data. We'll take a break. And again, if we're running ahead of schedule, we'll break early, but our intent is to start at the listed time on the agenda for the following set of presentations. Then we will go to real world data evidence, and then we'll have the Q&A session. We'll break for lunch, and then we will start at 1 o'clock with the afternoon sessions, which deal with what makes a high quality submission, adaptive study design, and adaptive study designs. And so at this point, we will transition. I would invite Aila Albrecht to the podium to begin her presentation.
Good morning. I'm Aila Albrecht. As part of this presentation, I'll provide an overview of the fees that are part of the Animal Drug User Fee Act, or ADUFA, review the 4 types of fees and how the fee rates are established every year, and go over the billing cycles associated with the fees. I'll also go over the 5 types of fee waivers that can be requested under ADUFA and share a list of publicly available resources with further information on ADUFA fees and waivers. ADUFA has 4 types of fees: a sponsor fee, an application fee, of which there are 3 subtypes, product fees, and establishment fees. Each type of fee is either a one-time fee or an annual fee and generates a certain percentage of revenue for the ADUFA program. I'll cover each of these 4 fees in more detail on the following slides. The first type of ADUFA fee is the annual sponsor fee. This fee is paid annually by each applicant who has a new animal drug application, or NADA, a supplemental new animal drug application, or an investigational new animal drug, or INAD, file. The term animal drug sponsor means either an applicant named in a new animal drug application or a person who has established an investigational new animal drug file. All sponsors, independent of the number of NADAs or INADs they hold, pay the same annual sponsor fee. Sponsor fees generate 27% of the ADUFA target revenue. The second type of ADUFA fee is the application fee, which is paid when an NADA or administrative NADA is filed with FDA. As the definition on this slide states, each applicant who submits an animal drug application or supplemental animal drug application shall be subject to an application fee. The full fee is for an original application, which is an application for approval of any new animal drug. There are 2 applications that trigger a 50% fee. First is a supplemental application that CVM classifies as a B1 supplement. B1 supplements require a reevaluation of certain safety or effectiveness data in the parent application. The second type of application that triggers a 50% fee is a combination application, which is a new application involving combination new animal drugs that have previously been separately approved for use in feed or water. Application fees generate 20% of the ADUFA target revenue. The third type of ADUFA fee is the annual product fee. This is paid annually by the applicant in a new animal drug application or supplemental new animal drug application for an animal drug product. The term animal drug product means each specific strength or potency of a particular active ingredient in final dosage form marketed by a particular manufacturer. Each NDC, or National Drug Code number, is assessed one product fee, which is based on the labeler code and product code in the NDC. Product fees generate 27% of the ADUFA target revenue. To review the parts of an NDC, the three segments of the NDC identify the labeler, the product, and the commercial package size. The first set of numbers in the NDC is the labeler code and identifies the labeler, such as the drug manufacturer, repackager, or distributor. The second set of numbers is the product code, which identifies the specific strength, dosage form, such as capsule, tablet, or liquid, and formulation of a drug for a specific labeler. The third set of numbers is the package code, which identifies package sizes and types. The final type of ADUFA fee is the annual establishment fee. This is paid annually by each applicant who owns or operates, directly or through an affiliate, a new animal drug establishment and is named as the applicant in a new animal drug application or supplemental new animal drug application. The term animal drug establishment means a foreign or domestic place of business which is at one general physical location consisting of one or more buildings within 5 miles of each other, at which one or more new animal drug products are manufactured in final dosage form. Establishment fees generate 26% of the target revenue. In order to estimate the number of fees FDA will collect in a given fiscal year, there must be some assumptions made. For application fees, FDA calculates the average number of applications over the 5 most recent completed years. For sponsor, product, and establishment fees, FDA uses a count based on the most recent May data in our systems. To determine the estimates for what percentages of fees will be waived, FDA uses the most recent 5-year average. In FY 2025, 68% of sponsor fees were waived, 2% of product fees were waived, and 6% of establishment fees were waived. FDA uses a variable inflation adjuster to calculate the annual inflation rate. This is done via a 2-step process. First, FDA calculates the latest 3-year average change in FDA personnel compensation and benefits, or PC&B costs, multiplied by the 3-year average change in proportion of FDA PC&B to total FDA costs. For FY 2025, the average change was 3.9%, and the average proportion of total costs was 50.8%. By multiplying these two numbers together, the first part of the inflation adjuster is 1.96%. Second, FDA calculates the latest 3-year average change in Washington, Arlington, Alexandria Consumer Price Index, or CPI, all items less food and energy, and multiplies it by the 3-year average change in proportion of FDA non-pay to total FDA costs. For FY 2025, the average CPI was 4%, and the non-pay costs were 49.2%. By multiplying these two numbers together, the second part of the inflation adjuster is 1.97%. The final step is to add parts 1 and 2 together for a total inflation adjuster of 3.93%. This percentage is applied to the base revenue and amount in statute. The base revenue amount in statute for FY 2024, the first year of ADUFA 5, is $33.5 million. The final 2 steps in calculating the fee rates are determining the workload adjuster, shown on this slide, and determining the operating reserve adjustment, if applicable, shown on the next slide. The workload adjuster is calculated using the average of five types of applications and submissions listed on the slide, received over the most recent 5-year period as compared to a rolling base of the most recently completed 5 years. Each application or submission type is multiplied by a weighting factor, which is the percent of direct review time spent on each of the 5 application and submission types. Therefore, the workload adjuster is driven by the change in key submission types as weighted by the number of hours those submissions take to review. The workload adjuster is not invoked unless it exceeds 3% for a second fiscal year in the authorization and any year thereafter. This means if it calculates at 2% for FY 2025 and 4% for FY 2026, it would not be added for FY 2026 because it did not yet exceed 3% for 2 fiscal years in ADUFA 5. Finally, FDA calculates the operating reserve adjustment. This provision allows FDA to increase or decrease the target revenue to ensure the program has a certain amount of funding in the carryover reserves. If the carryover reserve is less than 12 weeks, FDA will increase the fee rates to maintain a 12-week floor. If the carryover reserve is greater than 22 weeks in FY 2025, 20 weeks in FY 2026, 18 weeks in FY 2027, and 16 weeks in FY 2028, FDA will decrease the fee rates for that fiscal year to maintain these ceilings. In other words, the carryover reserve is part of the calculations when new user fee rates are set for each fiscal year. This reserve ensures FDA has an appropriate amount of funding on hand to manage situations such as a lapse in appropriations or under collections in a fiscal year. Listed on this slide are the FY 2025 ADUFA fee rates for the 4 fee types we reviewed earlier in the presentation. I'd also like to review some additional information regarding ADUFA fees. First, you may be familiar with the sponsor letter notifications. These Dear Sponsor packages are sent out every year mid-August prior to annual billing. This package includes a letter citing the new fee rates and links to the Federal Register notice, instruction page, and 3 verification reports. This process allows the sponsor to review documents and notify FDA of any changes prior to FDA sending out the initial invoices in December. ADUFA has 2 billing cycles, the initial billing and cleanup billing. Initial billing invoices are sent at the end of December for the current fiscal year, with payment due January 31st. This billing cycle includes all sponsors, products, and establishments FDA has listed in our database as of October 1st. Cleanup invoices are sent in November, and this includes all new sponsors, products, and establishments that were established after October 1st, but before September 30th of the prior fiscal year. To give an example of the billing timeline, in December 2025, invoices will be sent for everyone listed as of October 2025, and these invoices are due in January 2026. In November 2026, cleanup invoices will be sent for everyone listed after October 2025 and before September 2026. During the course of a year, cleanup invoices for the previous fiscal year come first, followed by standard invoices for the current fiscal year. So a sponsor may receive them 2 months in a row, one for the previous fiscal year, their first fiscal year, and one a month later for the current fiscal year. Unlike annual fees, where sponsors receive an invoice from the FDA, sponsors must proactively submit their application fees to FDA prior to filing their new animal drug application. The steps for paying an application fee include completing an ADUFA cover sheet, receiving a unique payment identification number that links the payment to the submission ID, paying the application fee, And finally, filing the application, including a copy of the cover sheet. The Federal Register notice, which details the annual fee rates and all the calculations described in this presentation, is mandated to publish 60 days before the start of the fiscal year, which begins on October 1st. These typically publish in late July or the beginning of August. FDA is also mandated to publish certain reports and plans for the ADUFA program. The annual financial and performance reports must publish 120 days after the close of the fiscal year and typically publish in late January. New in ADUFA 5 is a financial plan that was initially published in March 2024 and is updated annually with actuals for the completed fiscal years and projections for future fiscal years. These must be published 180 days after the close of the fiscal year and typically publish in late March. And now I'll provide an overview of the waiver options available to sponsors as part of the ADUFA program. There are 5 types of waivers. Sponsors may request waivers from or reductions of user fees under the following significant provisions. Following specific provisions: significant barrier to innovation, fees exceed costs, free choice feeds, minor use or minor species, and small business. In general, when FDA grants a waiver, it will be for the entire fee. The one exception would be for a fees exceed costs waiver. Sponsors may qualify for waivers from more than one user fee. However, they need to request one for each fee for which they are liable. The significant barrier to innovation waiver has 2 prongs to it: innovation and financial. Guidance for Industry number 170 describes in detail how we will make our determination as to whether the product is innovative. In terms of the second prong of the test, we review the sponsor's financials, namely their balance sheet, to make our determination. We review the sponsor's working capital, which is the value of their current assets minus current liabilities. For FY 2025, the inflation-adjusted amount is $23,258,600. So, in order for a sponsor to qualify under the financial prong, the sponsor's working capital must be under $23,258,600. For the fees exceed costs waiver, we cannot make our determination until after the fiscal year has ended. At that time, we calculate the hours spent on reviewing certain files and applications in order to develop standard costs for the review of specific submission types. The submission types are for the following technical sections: environmental impact, target animal safety, effectiveness, CMC, and human food safety. In addition, standard costs are used for administrative NADAs and supplemental NADAs with and without safety and effectiveness data. After performing these calculations, we determine whether a sponsor has submitted any of these 8 different component types and when. Standard costs are applied to these submissions and are compared to the amount of fees paid by the sponsor. If the amount of fees paid by the sponsor exceed the anticipated present and future costs incurred by CVM in conducting the process for the review of the new animal drug application for the sponsor, then the sponsor will be issued a refund. The free choice feeds waiver is available for a new animal drug application or a supplemental new animal drug application that is intended solely to provide for use of the new animal drug in a Type B medicated feed intended for use in the manufacture of a Type C free choice medicated feed or a Type C free choice medicated feed. When granted, this waiver typically applies to sponsor or application fees. A sponsor may qualify for a sponsor fee waiver only if their entire portfolio of files and applications is covered by a waiver. They may qualify for an application fee waiver if the application is intended solely to provide for use of the new animal drug in a free choice medicated feed. The minor use or minor species, or MUMS waiver, is for a new animal drug application or supplemental new animal drug application that is intended solely to provide for a minor use or minor species indication. As a reminder, a minor use means the intended use of a drug in a major species is for an indication that occurs infrequently and only in a small number of animals or in limited geographic areas and in only a small number of animals annually. Minor species are defined as animals other than cattle, horses, swine, chickens, turkeys, dogs, and cats. This waiver would apply to a sponsor fee as long as all the files and applications in a sponsor's portfolio are covered by a waiver. A MUMS waiver may also apply to application, product, and establishment fees. A small business waiver is granted to a sponsor involved in a small business submitting its first new animal drug application to CVM for review. A small business is defined as one that has fewer than 500 employees, including employees of affiliates. This waiver can be confusing for some in that it only applies to the first new animal drug application submitted by a sponsor. In other words, the waiver cannot be used for a sponsor fee. This slide shows a summary of the 5 types of ADUFA waivers and their applicability to the 4 types of ADUFA fees. There are 2 ways to request a fee waiver from FDA, electronically or in paper. We recommend submitting electronically as requests are received in a more timely manner. Using the eSubmitter tool, you will select ONAPE and OGAD submissions and document type General Correspondence, or G. For the 3 annual fees, sponsor, product, and establishment, we recommend requesting waivers at least 4 months prior to invoices being issued. As a reminder, initial invoices are issued at the end of December, so request a waiver by the beginning of September. And cleanup invoices are issued in November, so request a waiver by the beginning of August. However, the latest a waiver request can be submitted for consideration is 180 days after the fees are due. For example, waivers are due July 30th, 2025 for the FY 2025 invoices. For application fees, sponsors must submit their waiver request at least 30 days prior to submitting the application. This slide includes links to websites you may find helpful to review the information I've presented on today, including the main ADUFA page, the FY 2025 fee rates, the financial reports, the performance reports, and the financial plan. This slide includes links to guidances and websites related to waivers, including guidance for industry number 170 on ADUFA waivers and CVM's eSubmitter Resource Center page. And that concludes this presentation. Thank you.
All right. So at this time, we're going to move along to the next topic of the morning sessions on foreign data to support safety and efficacy. I'd like to invite Courtney Flick, Ana Lazo, and Brandi Robinson up to the podium, and they will begin their presentations.
Good morning. My name is Courtney Flick, and I am a veterinary medical officer in the Division of Companion Animal Drugs. Along with my colleagues Anna Lazo and Brandi Robinson, we're going to be speaking to you today about the use of foreign data. I will kick us off talking about ways that you can use foreign data in the approval process. Anna will explain about our expectations of foreign data submissions, and Brandi will bring us home with a discussion of our international collaboration programs. So we'll start with the basic question. What is foreign data? It's defined in Guidance 265. CVM considers foreign data to be data generated in studies that are conducted outside of the United States. The studies may be conducted by entities that are either based within or outside of the United States. So it's the location of the study itself rather than the location of the entity that commissions the study that determines if the data are considered foreign for regulatory purposes. FDA has a long-standing practice of accepting foreign clinical data to support applications. This was codified in the FD&C Act and reaffirmed in FDASIA. To be accepted, the data should be adequate under applicable standards to support approval. This applies to foreign data in submissions to investigational new animal drug files and new animal drug applications, including conditional approvals and generic animal drugs. But we're going to be focusing on pioneer drug approvals with this presentation. As a reminder, you are required to submit all foreign data from investigations or commercial marketing studies, regardless of whether the results are favorable or unfavorable. FDA is committed to supporting global approvals to enhance animal drug development, to facilitate the use of foreign data, and to minimize the need for duplicative studies. FDA also supports international harmonization activities that help leverage work and expertise from other regulatory authorities. CVM has accepted and will continue to accept foreign data for target animal safety, substantial evidence of effectiveness, reasonable expectation of effectiveness, and for the human food safety technical sections. Foreign data may be used to provide supporting safety and effectiveness information, and to bridge gaps in U.S. data. CVM will determine whether the data fulfills our needs, both in terms of study conduct, documentation, and how well the data can be used to inform a U.S. regulatory decision. We remind you that foreign data or information not used directly to support an approval should also be submitted in the All Other Information technical section. That includes information derived from foreign markets and summaries of foreign adverse event experiences. Guidance 185 outlines the requirements for the evaluation of target animal safety. These guidelines pertain to studies conducted in the United States or at a foreign site. We recommend you discuss with CVM how a foreign study may deviate from these guidelines, ideally prior to performing the study or before submitting the data. Guidance 226 outlines the recommendations for the submission of safety data to support approval of a new animal drug. These are some common challenges we see when reviewing data from foreign safety studies. Regulatory compliance refers to a study not conducted in compliance with U.S. GLP regulations. Whether you're planning to submit foreign data to support target animal safety technical section, or if you're planning to conduct a safety study outside of the United States, we recommend you discuss the data and/or study design with CVM prior to submission and submit the protocol for CVM review. I will be providing several examples where foreign data were used to support a technical section of an approved product. You may refer to the FOI summaries for these approvals for more detailed information. So this slide has examples of approvals where target animal safety was supported solely by foreign data. For Aquaflor, for the supplemental approval for the control of mortality in freshwater reared salmonids, both margin of safety studies were conducted in Scotland. For Iradia, for the approved for the treatment of Giardia duodenalis infection in dogs, the laboratory margin of safety study was conducted in France. For this example, the study differed in regulatory compliance and was conducted following OECD principles of GLP rather than US GLP regulations. Anna will discuss in more detail what to do when a study is conducted under a different regulatory standard. These examples are cases where foreign data was used in conjunction with domestic data to support target animal safety. For Safeguard Aquasol, for the treatment and control of Escherichia galli and Heterachus gallinarum, the reference interval study and 2 margin of safety studies were all conducted in the UK, while the reproductive study was conducted in the United States. For Varenzin CA1, conditionally approved for the control of non-regenerative anemia associated with chronic kidney disease in cats, the laboratory margin of safety study was conducted in Germany, again under the OECD principles of GLP. For conditionally approved products, we often pull additional safety information since the field safety evaluation has not yet been conducted. In this case, safety was also supported with the results of a laboratory PK/PD study also conducted in Germany, as well as an interim field safety analysis from the in-progress pivotal field study being conducted in both US and European sites. For Upcard CA1, conditionally approved for use with concurrent therapy for the management of pulmonary edema in dogs with congestive heart failure, safety was supported by a 13-week margin of safety study conducted in Scotland with a tablet formulation of torsemide. A second non-terminal 6-month GLP laboratory safety study was conducted in the United States. Field safety was supported by the results of 2 field effectiveness and safety studies conducted in Europe with the tablet formulation. The results of the European studies were bridged with a pharmacokinetic relative bioavailability study comparing the European tablet formulation to the US oral solution formulation. The Division of Human Food Safety routinely reviews data generated from generated in foreign countries, and there are many cases in which foreign data have been used to complete the human food safety technical section requirements. The expectation is that studies are conducted in accordance with appropriate VICH and OECD guidelines and conform to GLP standards. You can find more information about the use of foreign data to support human food safety in the data quality webinar linked on CVM's website. For CVM to consider the use of foreign investigational studies to support the effectiveness of new animal drugs, the studies should have the same data quality and study integrity standards as expected of domestic studies. Guidance 265 describes situations where data from foreign investigational studies may be considered acceptable to support effectiveness requirements. The guidance also describes the avenues by which a sponsor may obtain feedback from CVM on the acceptability of foreign data and on international collaboration opportunities. Brandi will talk more about those opportunities later in this presentation. This slide describes some factors that may limit the acceptability of foreign data to support US regulatory decisions. These points should be considered when either planning a foreign effectiveness study or justifying the acceptability of previously collected foreign effectiveness data. There are other considerations that are not captured here based on the population, the study design, or other differences. If gaps exist in those factors, you should propose ways to bridge those gaps prior to the data submission in a meeting or in an H submission. You may also submit that information in the technical section with the submission of the study data. These are some potential challenges that may be encountered when collecting foreign data or using previously collected foreign data to support effectiveness. Differences in the indication or drug characteristics may impact the study design and affect our ability to accept a study for substantial evidence of effectiveness. As with target animal safety, we recommend that you talk to CVM when considering the use of foreign data to support effectiveness. These are some examples where the effectiveness evaluation was based solely on foreign data. For Sileo and Pexion, both of these drugs are approved for the treatment of noise aversion in dogs. Substantial evidence of effectiveness was demonstrated with a European field study in both cases. The European studies used a noise event, which was New Year's Eve fireworks, which is comparable to fireworks in the United States. Banamine transdermal solution for the supplemental approval for the control of pyrexia associated with acute mastitis in dairy cows. Substantial evidence of effectiveness for this approval was also supported with a multi-site European field study. These are examples of food animal drugs where effectiveness was supported by data collected both within the United States and in foreign sites. VICH GL7 allows CVM to accept one of two dose confirmation studies conducted in a foreign country, as long as the animals and the management practices are comparable to the United States. The VICH also allows for both dose confirmation studies to be conducted outside of the United States when the parasite is not common enough to run the study in the United States. For long range, approved for the treatment and control of a variety of internal and external parasites in cattle, this example highlights both of the situations outlined in the VICH. For the endoparasitic indications, there are multiple situations where one dose confirmation study was conducted in Europe. And for the Sarcoptes field study, we accepted European sites as well. For Rumensin, approved for a variety of antiparasitic indications, in the 2004 supplemental approval for increased milk production efficiency, and for other later supplemental approvals, the effectiveness studies were conducted in both the US and Canada. These are examples of companion animal drugs where effectiveness data was collected within the United States and also at foreign sites. For Librela, approved for the control of pain associated with osteoarthritis in dogs, the sponsor conducted both US and European field studies to support effectiveness in the two jurisdictions. While the US and European studies had similar success rates in the treated group at an early time point, the US study had a higher success rate in the control group. So the difference between treatment success rates between the treatment and control groups was not significant at that time point. The sponsor was able to leverage the European field study data and use a weight of evidence approach using to demonstrate substantial evidence of effectiveness for the US approval. For Aradia, approved for the treatment of Giardia duodenalis infection in dogs, effectiveness was supported by one laboratory dose confirmation study conducted in the United States and one multi-site field trial conducted in Europe. CVM accepted the European field study because Giardia found in dogs in Europe is indistinguishable from the parasite found in the United States. For Duotic, approved for the treatment of otitis externa in dogs associated with susceptible strains of yeast, the field study to support substantial evidence of effectiveness enrolled cases from sites both in the US and Spain. For Credelio Quattro, approved for a variety of ecto and endoparasitic indications, the laboratory studies supporting the effectiveness for intestinal roundworm indications were conducted in South Africa and Morocco, in conjunction with U.S. laboratory and field effectiveness studies. That's all from me. I'm going to hand things over to Anna to talk to you about our expectations of foreign data submissions. Thank you.
Thank you, Courtney. Good morning, everyone. My name is Ana Lazo, and I'm a Quality Assurance Study Reviewer in the Division of Business Information Science and Management. Today, I'll be presenting on CVM's expectation for foreign data submissions. Let's start off with communication. CVM encourages sponsors to explore a variety of design options in the planning of studies and therefore highly encourage early communication with us. There are various approaches that sponsors may take to open a discussion on the use of foreign data. For example, this can be done through a meeting request or a technical section submission. An organized and focused information package allows CVM the opportunity to provide you with appropriate recommendations. Therefore, as with any study submitted to CVM, when presenting foreign data, please assure the following elements are addressed. As previously mentioned, all studies should have or will have the same data qualities and study integrity standards as those expected from domestic studies. The submission should explain how the results of the foreign study will be applied to the target population within the United States. Clearly tell us the standard of conduct followed or that you plan to follow, and make sure to discuss the impact of any differences in the standards followed if different from FDA standards. And most importantly, tell us how the study will fit within the overall development plan. As with any study submitted to FDA, data is generally assessed, evaluating several factors, including but not limited to those outlined on the slide. Specifically for studies supporting non-clinical laboratory studies, 21 CFR 514 1B12 justifies why we have for a very long time required these to be conducted in compliance with 21 CFR Part 58. If a different standard is chosen, then you must determine what aspects of the study were not in compliance with 21 Part 58 and explain its impact on the compliance statement. So remember, the GLP compliance statement should include a discussion on any areas of noncompliance and the reason why 21 CFR 514 or CFR Part 58 were not followed. This would be true for OECD studies, as well as any other study that did not use US FDA GLPs. Continuing with sponsor GLP compliance statement expectations. A sponsor GLP compliance statement should be provided for each GLP study. It should affirm the study's compliance with 21 CFR Part 58 or provide a statement with a reason for the noncompliance. It should include all critical deviations and GLP exceptions noted in the final study report with the impact assessed. As previously mentioned, for studies conducted using standards other than FDA GLPs, describe how you did not comply with FDA GLPs. For example, if an item of noncompliance is consistent with OECD GLP but not FDA GLP, the sponsor should explain the difference between the two standards of conduct for your particular study and the impact of the noncompliance on the study. The sponsor may provide the differences between OECD GLP and FDA GLP as part of their explanation of noncompliance, but this is not required, nor is a statement or a chart of differences alone sufficient. Fully describe how the study complied with FDA GLPs and why the noncompliance to FDA GLPs did or did not affect the outcome of the study results. This statement should be signed and dated by the sponsor representative responsible for making these assurances. You can refer to the questions and answers document for the data quality webinar updated April 2021 for a few examples of sponsor compliance statements. Some examples of compliance statement challenges noted during our review are missing sponsor compliance statements, or the statements do not include all items of noncompliance. In addition, the sponsor does not fully assess or provide an impact statement for each of the items of noncompliance. Again, providing the differences between the standard of conduct selected to conduct the study and 21 CFR Part 58 is not a sufficient assessment and explanation. We need to fully understand the specific noncompliance to FDA GLPs that occurred for your study and what impact it has on the quality of your data submitted to CVM. Moving on to raw data expectations. During data collection, it is important to keep in mind that all units of measure should be consistent with the imperial system of measurement, for example, inches and pounds, with the international system of units, SI units, or acceptable SI derivatives, for example, centimeters and kilograms, or a combination of both as appropriate. Raw data should be submitted as recorded. However, all data for a specific variable should be converted to the same unit of measurement for evaluation and documented in the final study report. The final study report should also include full conversion details. Moving on to use of foreign data and privacy concerns. Studies in compliance with GCP and GLPs are sometimes conducted at facilities located outside the United States, therefore introducing privacy concerns due to local laws. We acknowledge that sponsors have raised concerns with meeting their local privacy requirements, but we'd like to remind you that drug companies are responsible for ensuring all data from foreign studies are compliant with local laws while still maintaining the requirements for approval of new animal drugs in the U.S. CVM recognizes that foreign privacy laws may pose challenges in the submission of data to CVM. Therefore, we recommend that sponsors consult with their attorneys and/or consultants of their locality to ensure that EU investigators and/or consultants are aware of the studies that support approval in the EU and US are aware of this requirement. One way that you can do this is by obtaining consent from their EU investigators to provide this information to us, either by adding it to their contract or study protocols. One example of a challenge that we typically see upon submission to CVM is that CVs of study personnel or training documentation for protocols, regulations, and/or electronic data collection systems are not provided. Without this information, reviewers cannot confirm whether the study personnel had adequate training or were qualified to perform their assigned functions. As a reviewer, we must be able to confirm that study personnel received adequate training specific to study protocols, the EDC systems they are using, and continuous refresher training on GLP and GCPs. Translations. If any part of the application is in a foreign language, an accurate and complete English translation of each part that is not in English shall be appended. Therefore, in order to meet this requirement of FDA regulations, translation of study data and study reports may be necessary. Our expectations are that drug companies will ensure that foreign studies meet the requirements for approval of new animal drugs in the U.S. We'd like to refer you to CDER's draft guidance, Translation of Good Laboratory Practice Study Reports, Questions and Answers, for more information on requirements for GLP study reports. In contrast from the previous slide, this draft GFI states that GLP study reports should include a certified translation. In addition, every translated study report should be certified for accuracy. Certification should come from the translator or a qualified third party, and a certification statement must confirm that the translation is accurate and complete to the best of the translator's knowledge. It also has to maintain consistent terminology across translations, especially for technical and regulatory terms. Using standardized industry terms is crucial to avoid confusion. As part of the submission process, translated reports should be included as part of a regulatory submission. When submitting electronically, ensure that the translation file is clearly marked and distinguishable from the original report. Finally, include a translator certification with the report when submitting to FDA. Overall, this GFI aims to ensure consistent and accurate submission of reports in different languages. During our review, we routinely notice challenges related to translations. Upon review of the study data, we find that not all data recorded in the original foreign language were translated. We find that only sections of data may be translated. For example, we find that dates or study participant names and roles are not translated. We therefore cannot identify the individuals, confirm proper training, or determine if the correct individuals are performing the assigned tasks. In addition, we also find that data may not always be recorded in the original language. For example, the observer may call out an observation to a recorder that a person translates and records the data in English. This translation may not always be correct or mean the same as the original observation. For example, this can be an observation of an animal that is favoring a paw. This may be translated and recorded in English as an animal limping, which is not the same. We encourage that the individual translating data be knowledgeable and have familiarity with the data to assure accurate translations. To help us ensure that we can depend on data submitted to support a U.S. approval, we rely on the Bioresearch Monitoring Program. FDA may conduct inspections of clinical investigators and non-clinical studies in support of U.S. drug approvals or utilize inspections from regulatory agencies in the relevant country. Through an inspection, BIMO can verify the accuracy and reliability of study data submitted to FDA in support of an application. To be clear, FDA would only inspect to FDA GLPs, but regulatory agencies in other countries will perform their inspections to their own standards, assessing the clinical investigator and the facility compliance program. For more information, we'll refer you to the BIMO compliance program. One question that CVM was asked is how do sponsors increase the likelihood that data initially generated for a foreign dossier is accepted as pivotal by CVM and can support substantial evidence? When submitting foreign data already collected to the FDA, it's imperative to present this data in the context of a U.S. approval. That is, FDA expects that the sponsor will present the data in a way that supports the proposed indication, dosage regimen, and conditions of use being pursued in the U.S. Another question is how do sponsors increase the likelihood that data generated for both a foreign dossier and CVM will be accepted? Well, we encourage you to have early communication with us regarding the study design, including how the proposed population is representative of the target population in the U.S. We recommend that you submit a protocol for review and advise that you consider one of the international collaboration options that you will be hearing about immediately after this presentation. With that, I conclude this presentation. Thank you for your attention, and I will now pass it along to Brandi Robinson.
Thank you, Anna. I'm Brandi Robinson, and I serve as the International Program Manager for the Office of New Animal Product Evaluation here within the Center for Veterinary Medicine. In the coming slides, I'll be sharing information on our international collaborative review opportunities. These opportunities offer a proactive means to ensure foreign data can fulfill pivotal data needs for FDA approval. Our collaborative review opportunities are made possible by two key components. The first is CVM's participation in the International Cooperation on Harmonization of Technical Requirements for Registration of Veterinary Medicinal Products, also known as VICH. My fellow presenters have already mentioned VICH this morning. The other component that serves as a foundation to our regulatory collaboration opportunities are the confidentiality agreements established between FDA and many of our regulatory counterparts. These agreements allow us to discuss confidential information with our regulatory counterparts with the assurance that such information will be protected from disclosure. All confidentiality agreements are available on the FDA website. As I mentioned, VICH has already come up earlier today, but I'd like to give a little more background and context to how our participation in VICH opens doors for greater regulatory collaboration. VICH develops harmonized guidelines on the technical requirements to support safety, effectiveness, and quality of animal drugs. CVM is one of the founding members of VICH, and we implement VICH guidelines as FDA guidance for industry. The guidelines are harmonized with the European Medicines Agency and the Japanese Ministry of Agriculture, Forestry, and Fisheries, as well as other regulators who choose to implement the VICH guidelines. Harmonized guidelines reduce the need to conduct multiple studies to meet the regulatory requirements in multiple regions, reducing the cost to develop animal drugs in a global market. Our harmonized guidelines and our confidentiality agreements mean that we have similar standards to our regulatory counterparts and the ability to discuss non-public information. So when I'm speaking about our collaborative review opportunities, I'm referring to opportunities for a sponsor to work with both CVM and another country, region, or multiple countries at the same time for a product under review. We have three main options available at this time, and they vary in scope and the level of commitment. We have a simultaneous review option between CVM and Health Canada's Veterinary Drugs Directorate, or VDD. We have parallel scientific advice available with the European Medicines Agency, EMA. And our most recent option is still in a pilot stage. This third option is a 5-way collaborative review involving CVM with the Australian Pesticide and Veterinary Medicines Authority, APVMA, New Zealand's Ministry for Primary Industries, MPI, the UK's Veterinary Medicines Directorate, VMD, and Health Canada's VDD. Our simultaneous review program with Health Canada's VDD began around 2011. This collaborative review opportunity allows a sponsor to follow CVM's phased review process through the full review process with both CVM and VDD. In the context of using foreign data, this tool offers the opportunity to discuss studies with two regulators to ensure the locations of those studies will be acceptable to both. A sponsor might consider conducting studies in Canada, the US, or both. They could also discuss the acceptability of conducting studies elsewhere to meet the US and Canadian requirements. This pathway gives a sponsor a proactive opportunity to ensure the same fundamental data sets will be acceptable and reviewed simultaneously in the US and Canada. If a sponsor is interested in simultaneous review between CVM and VDD, they would request to add their project. If the project is accepted, the sponsor agrees to submit the same information to both agencies at the same time. Each agency reviews the information independently. The reviewers can discuss their respective reviews, and each agency provides its own responses. It is important to note that each agency will follow all applicable regulations and policies in their jurisdiction, and that specific regulatory decisions may differ. This program offers the opportunity to follow the full review process towards approval in both the US and Canada at the same time. Another excellent opportunity to proactively discuss foreign data comes with the Parallel Scientific Advice Program. In this opportunity, a sponsor can submit a series of questions to the European Medicines Agency and CVM at the same time. These are questions you would typically ask in a pre-submission conference and could relate to specific issues you are considering in study designs. Unlike our simultaneous review program with Canada, this is a single point of collaboration. CVM and EMA recently revised this program and have documents on our website containing more information. As with our simultaneous review program, each regulator will follow their respective laws and regulations. The aim is to align our advice to the extent possible and to explain any differences if they exist. The documents on our website contain some points to consider to support a successful parallel scientific advice request. These include ensuring a high-quality submission, ensuring that the product is intended to be approved and authorized in each jurisdiction, and the documents contain some pointers on the types of questions which could be included. This is not intended to be an exhaustive list. One of the biggest changes in the revised program is that it now includes a trilateral meeting with EMA, CVM, and the sponsor. We have one last collaborative review tool that I'd like to share this morning. This option is still in the pilot phase. If a sponsor has the same product approved in the US, Canada, Australia, New Zealand, and the UK, and is seeking a supplemental approval in all 5 countries, we have the opportunity for a sponsor to submit the application to all 5 jurisdictions for review. This program requests the same information be submitted to all 5 regulators. The information is then reviewed, and the comments or questions from all 5 regulators are consolidated into a single request to the sponsor. As with our other options, each regulator will make its own decision based on its laws and regulations. If anyone is interested in more information or in utilizing any of our regulatory collaborative review programs, please feel free to contact me. These all offer an excellent opportunity to discuss the possible use of foreign data proactively in your animal drug development plan. We have several slides here with resources you can consider. These are available on our website. This first one includes some of our Q&A documents. This slide includes some of the guidelines that were referenced this morning, including some of our VICH guidelines. And this is a link to some of our resources related to regulatory collaborative review with our counterparts. And with that, I will hand it over to Walt. Thank you.
Thanks, Brandi. So at this time, I will let you know we are running ahead of schedule. And so we will take a break and we will resume at 11 o'clock, as mentioned in the agenda that's been put out. So we will be back at 11 o'clock. Oh, one other thing. By the way, we've been receiving questions through the QR code. Don't hesitate to send the questions in, and you can submit your question at any point during the day, regardless of the topic of your question. So if you're asking questions about this morning's topic, but then you say, wait a minute, this afternoon I've got another question to ask associated with it, feel free to ask that. So again, we'll see you at 11 o'clock. All right, welcome back. We will proceed to our next presentation. Dr. Emily Smith will be presenting on real-world data and real-world evidence to drug support effectiveness in new animal drugs.
These words are tongue twisters, so I'll try to speak slowly and not mess them up. Good morning, I'm Emily Smith, and I'm a veterinary reviewer in the Division of Food Animal Drugs. So our objectives today with this presentation are to establish an understanding of the regulatory definitions related to real-world data and real-world evidence, and the key questions that you want to answer when you're evaluating whether real-world data and real-world evidence may be appropriate to support the approval of new animal drugs. We're also going to provide you at the end with resources for where to look for more information if this is an approach that you're interested in pursuing. So in order to understand the context of the regulatory application of real-world data and real-world evidence, we need to start with the law that underlies these definitions. So Section 305 of the Animal Drug and Animal Generic User Fee Amendments of 2018 required FDA to issue guidance, and I quote, "For the purposes of assisting sponsors in incorporating real-world evidence, including ongoing surveillance activities, observational studies, and registry data, into proposed clinical investigation protocols and applications for new animal drugs." The law focuses on effectiveness and thereby submissions to investigational new animal drug files, new animal drug applications, and applications for conditional approval. For new animal drugs, the use of real-world data and real-world evidence could apply to both original applications as well as supplements. And we'll discuss some of the more likely applications of this type of data later in this presentation. Now, the law for veterinary drugs was passed about 2 years after the 21st Century Cures Act. And the 21st Century Cures Act both provided a definition for real-world evidence for human drugs and biologics, as well as it established a mandate for the real-world evidence program. So you'll see some carryover between the terminology in the 21st Century Cures Act and also the ADUFA-AGDUFA law. So as required by the law in 2018, we published Guidance 266, and that was titled "The Use of Real-World Data and Real-World Evidence to Support Effectiveness of New Animal Drugs." In this guidance, we provide definitions of both real-world data and real-world evidence as they apply to veterinary drugs. So our definition of real-world data are data routinely collected from a variety of sources relating to the health and productivity of animals, the delivery of veterinary care, or the management of livestock for food. Real-world evidence is the clinical evidence of the effectiveness of a new animal drug, and that is derived from analysis of real-world data. And as you can see, because the focus of the law and the guidance is on effectiveness, these definitions focus on effectiveness, but this would not preclude the use of real-world data and real-world evidence to support safety. We also want to emphasize that we issued this guidance at the time, this was in October of 2021 when we issued the final guidance, based on the developing regulatory science and not based on lots of approvals and extensive experience having used real-world data in the veterinary context. Many of the recommendations and techniques for collecting and analyzing real-world data have been expanded and developed on the human side through recent guidances. But we recognize that the sources of real-world data for veterinary drugs are different, and some of the challenges that we're going to experience in collecting and analyzing that data are also going to be different. So we're really going to need to work together to find ways to use this valuable information in the support of the approval of new animal drugs. Before discussing how we're going to use real-world data and also use real-world data to generate real-world evidence, we need to clarify the distinction between real-world data and clinical study or research data. Now, it's easy to be confused by these terms because many of our veterinary studies are conducted at clinics or farms that are simulating actual conditions of use. But what makes real-world data distinct from clinical research data is that it is obtained through routine veterinary care in animal management outside of a research setting. So in this case, if a drug is being administered in this context, the drug is being administered according to the veterinarian's clinical judgment. So if there's multiple drugs available to the veterinarian, they're going to administer the drug that is appropriate for that animal's particular clinical condition. By contrast, clinical study data or research data are those data collected according to protocol-specified procedures, and they are being conducted for research purposes. And in this context, drugs that are administered based on the assignment of an animal to a treatment group according to a research protocol. And these distinctions seem very clear, but in the real world, there are some gray areas. And one example of this is study designs where a clinical research study could incorporate some data from routine health care settings, or even mobile devices capturing health data that is outside the research setting. So in the next series of slides, I'm going to walk you through some of the key questions that should be answered for any project where you may be considering the use of real-world data and real-world evidence. We cover these topics in detail in Guidance for Industry 266, and there's some additional resources covering particular aspects of this on FDA's real-world evidence page. But we want to give you kind of a high-level overview and kind of walk you through these questions. Hopefully this will help you also anticipate some of the challenges that you might experience if you're considering the use of this type of data. Answering each of these questions is really important to ensure that the data and potential evidence that's generated is going to be able to answer the regulatory question that you're proposing it for. We also put these questions in a particular order because we think that this order will, walking through the questions in this order will help to improve data quality and integrity and maybe minimize some of the types of bias that's also, often experienced when in the real-world data context. So the key questions that we're going to cover are first, what is the regulatory question and purpose that you're proposing to use the data for? Secondly, what sources of real-world data are available? We're going to focus these sources on the veterinary, for veterinary drugs. Does the data have sufficient fitness for use? And by fitness, I mean relevance and reliability. And we're going to define those terms. Is a study using real-world data appropriate to answer the regulatory question? There are situations where you may use real-world data, but you're not actually using it in a study. What type of study designs are appropriate if you are conducting a study using real-world data? And if you are doing a study, what do you need to include in your protocol? So the first and we consider the most important question for you to answer is, what is the regulatory purpose for which you're using the real-world data? And it's this question that then determines the sources that you may select and a study design. So we've simplified the potential regulatory purposes into 3 separate categories. And these categories represent increasing reliance on real-world data. So the first is using real-world data to support a product development plan, and this may include characterizing a dosage regimen or defining conditions of use. The second category is use of real-world data for protocol development for a traditional clinical study. And in this situation, you may use real-world data, for example, to justify your enrollment criteria or your sample size for this traditional clinical research study. And third, which is the highest level of reliance on real-world data, is the use of real-world data to generate real-world evidence to demonstrate substantial evidence of effectiveness or reasonable expectation of effectiveness. So once you've defined your regulatory question, then you need to explore the data sources. The sources of real-world data for veterinary product development are outlined in more detail in our guidance. But briefly, the sources that are available include health and management records, and this would include paper or electronic records that you may get from a veterinary clinic or a production facility. And these are going to be probably your most widely available source. Other potential sources include pharmacy records, data from disease surveillance programs, product and disease registries, digital health, and this would include like wearable monitors, collars, ear tags, biosensors, and insurance claims. We mentioned pharmacovigilance data on this slide, but it's in italics because it's not a very useful source of information for effectiveness. The real-world data in any pharmacovigilance databases that relates to effectiveness would be extremely limited. Our current veterinary systems are mostly passive. Hopefully we'll have some active systems that are more widely available in the future. So it's voluntary reports of adverse health events. And we also get product quality complaints, defects, lack of effectiveness. And this information is great for signal detection, early warning, hypothesis generation, but not going to be useful in this context for generating data to support effectiveness. Once you have potential source or sources, then you need to evaluate the fitness of the real-world data and the source. And now fitness is divided into two components, relevance and reliability. So the first, evaluating whether the real-world data and the source has sufficient relevance, you need to answer whether that source is adequate for the regulatory purpose. This will include evaluating whether the source captures all of the elements that you actually need to analyze. So if you are conducting a study to demonstrate effectiveness, does this source even have all the information you need to conduct your analysis? Does it have the outcomes that you need to measure for effectiveness? Does it sufficiently capture the drug that's, information about the drug that's being studied? You also need to look at the standardization in that data collection. In some cases, the data may be collected using certain terminology that then have to be translated into a common set of terms in order to analyze it. Evaluating whether the animal population that's in this data source is representative of the animal population for which the drug is being intended. And now, are there a sufficient number of animals in the data source for the purpose that you're intending to use it? In a traditional study, this would be like a sample size estimation. And also evaluating if you need supplemental data sources. So in some cases, a single source will not have all the information you need to answer the regulatory question. So you may need to combine or link two sources together. And there are things you need to look at to determine whether it's even possible to link those two things together. In the real world data context, we refer to that as interoperability. The next question related to real world data fitness is reliability. And reliability is determined by the quality and integrity of the real world data and the source. So this reliability assessment first involves an evaluation of how the data are collected within that proposed source. We call this data accrual. And because real-world data are not originally being collected for research purposes, but they're captured as part of the normal care and monitoring of animals, we don't expect this data to be as tightly controlled as a clinical research study. But it does have to have sufficient reliability for the purpose for which you're intending it. We also think it's important to make sure that you're considering reliability throughout the data lifecycle. And this means assessing the quality and integrity from the source all the way through to the final data set. And there's a lot of steps involved between the source and the final data set, and we'll get into that in a little bit. So the data quality characteristics that you're looking for are similar to traditional clinical studies. High quality data are attributable, legible, contemporaneous, original, and accurate. So you recognize the acronym ALCOA. In a real-world data context, you will need to have some examination of the source to ensure that this data are of sufficient quality for the purpose that you're using it for. This may include the ability to verify information from the source and look at the timeframes over which it was collected. And you're going to generally have more accurate data when sources have standardized processes for data entry or have established clinical definitions. This will allow you to extract information from the source without as many steps into that analytical data set that could introduce errors. The data integrity assessment includes evaluating for completeness. This involves assessing for missing data, which can be a big problem in real-world data sets. Consistency and plausibility is evaluated for the data integrity assessment of accuracy, looking at provenance and traceability. If you have found sources that have appropriate fitness to answer the regulatory question, then the next thing you want to look at is whether a study using real-world data is appropriate to answer the regulatory question. Now, a study using real-world data is generally not the first choice. So if you have a project where a traditional clinical study is randomized controlled study, is feasible and ethical, that's still the gold standard for generating substantial evidence of effectiveness. We don't consider a study that's using real-world data to just be a one-to-one replacement for a traditional clinical study. However, there are definitely situations where a randomized controlled study is neither feasible nor ethical. One example of this might be a disease that has high mortality and there is not an appropriate active control for a comparator. And we also recognize there's situations where the use of real-world data could potentially increase the generalizability of the data to your intended population. But if you've determined that a study using real-world data is appropriate, there are some therapeutic areas where the analysis of effectiveness is likely going to be more interpretable and be able to overcome things like confounding and bias, which we'll define those on the next series of slides. These include indications where a large treatment effect size is expected, and this helps, again, like I mentioned, to overcome additional analyses to address confounding and bias. Diseases where you have an objective and a well-defined effectiveness outcome that is recorded in that real-world data set. And diseases with a predictable natural history, because this will allow you to create an appropriate comparator for your real-world data set. If you've determined that a study is appropriate, the next question is, what type of study are you going to use? And there are a variety of different study designs available using real-world data, and they have a variable reliance on that data. It's important that you select a study design that's most appropriate to answer your regulatory question, rather than selecting a study design that may just fit a particular data source that might be available. So this could be a limiting factor. You may not have, you pick the appropriate study design, but you may not have a data source that matches that well. A couple of examples that we want to share with you that represent potential options of study designs. First is a single arm study that would use real-world data as the external control arm. So in this particular type of study, the treatment arm is including clinical study data, but the comparator using real-world data, the comparator is using real-world data. So this is a non-randomized interventional study. The second example that we wanted to share is a non-interventional, non-randomized study, or we call it an observational study. And this is a type of study that has the highest reliance on real-world data. And this could include either prospective collection of real-world data or retrospective collection of real-world data. In these types of studies, the real-world data are compared to either historical control or a performance goal. And a performance goal is a specific numerical value that has been determined to be sufficient to demonstrate effectiveness. If you're conducting a study that uses real-world data to establish substantial evidence of effectiveness, the study must be adequate and well-controlled. And the protocol should include all the elements that are required by our regulations in 21 CFR 514.117. We discuss how to apply each of these elements to a study using real-world data in our guidance. And I'd like to share with a few of those elements today and how those would be reflected in your protocol. As for a traditional clinical study, ensuring for effectiveness, ensuring the study is conducted using an acceptable standard of conduct is important. Typically, that's going to be good clinical practice. And you can apply the concepts in GFI 85 to studies that are using real-world data, including observational studies. The study objective needs to be clearly defined, and I'm going to give you a little acronym on the next slide that helps to define your study objective for especially observational studies. Your protocol should include a discussion of the sources that you're using and a justification for fitness for use. And as we discussed earlier in the presentation, that this includes both relevance and reliability. So you explain why these particular sources have appropriate fitness. Your protocol should explain the procedures for data processing and how you're going to prepare the final real-world data set from the source to the analytical data set. Identify the potential sources of bias and confounding and how you're going to control for them. data checks that you'll perform on your real-world data, your statistical analysis plan, and the criteria that you're going to use to determine the effectiveness of the drug. So we call this the basis of study conclusion. So for an effectiveness study, you can define the objectives using the acronym PICO, which is a widely used acronym that's for research questions for observational studies. So the objective should be the evaluation of the effectiveness in a defined population. This will be your target animal species and any classes that it's applying to. This is for a particular drug product called the intervention based on comparison to a appropriate control using an endpoint that'll be analyzed to make the effectiveness decision. Now, you can see that this intervention is a particular new animal drug. If you're conducting a study for substantial evidence of effectiveness, it is your drug product formulation, unless there's other linkages that are being made. And so then on the next few slides, we're going to discuss how you take your real-world data and you prepare, analyze, and interpret that data. And I know that was one of the questions that was asked from last year in the educational conference. So real-world data sets are created by extracting individual animal data out of the source and then processing them into a data set for analysis. We call this process curation. The plan for how you're going to curate your data set needs to be described in your protocol, and that process then fully documented in your study file. And this processing involves a variety of considerations that will depend on the data source. This includes things including the time frame that you're going to extract data. So your source may have 15 years, and are you going to extract all of that time frame, or are you going to extract a certain period of time? And that decision will need to be made depending on the question that you're asking. A plan for how you're going to handle unstructured and structured data if it exists. So an example of unstructured data would be handwritten notes in a veterinary file, and those would have to then be transcribed in a way that then can be put into an analyzable data set. So as you can see, if you have unstructured data, that could create additional steps that you have to take in order to get to your analyzable data set. So if you have an electronic data set with standardized clinical definitions, that's going to make things a little bit easier for you. You need to have a plan for how you're going to link data sources if you need them. So maybe your one data source doesn't have everything that you need to conduct your analysis. So you have to put two pieces of data together. One example of this might be a veterinary medical record that has to then be paired with lab results that are coming in. And those lab results may not already be integrated into the veterinary medical record. So you have to have a way to link those together in a way that is not introducing errors into the data set. You may also have to standardize data. If you're collecting data from multiple veterinary clinics, for example, they may not all be using the exact same terminology across their veterinary records. And so there needs to be a pre-specified plan for how you're going to classify certain elements of that data in order to appropriately combine them into a single data set. And they may not even be in the same format. So you can start to see some of the challenges that might arise depending on the sources that you have available. Your protocol is also going to need to identify sources of bias and how you're going to control them. And I'm going to mention a couple of sources of bias here. A common source is selection bias, and this is where the study animals chosen for the study may not be representative of your target population of interest. And this would lead to results not being generalizable to the target population. And so you should make sure that you're selecting from all available sources to avoid selection bias. And when you're assessing your real-world data sources, making sure that those animals are representative of the animals for which the drug would be demonstrated to be effective. Another example is information bias, and this comes from measurement error in the data. Measurement errors in the exposure, the outcome, covariates. And you can minimize this type of bias with well-designed evaluation of endpoints and outcomes and the use of objective outcome assessments. Confounding can lead to the false conclusion that there's a causal relationship between treatment and outcome when there's really only a correlation between them. And so one real world example is where a veterinarian might be more likely to prescribe a new drug to animals with severe forms of a disease, the severe disease being the confounder. So those animals may be more likely to have a negative outcome than animals with milder disease. So within your real world data set, the new drug actually might be associated with worse outcomes, even if the drug's really beneficial or simply has no effect. So in our traditional clinical studies, bias and confounding are reduced by things such as masking, randomization, strict animal inclusion-exclusion criteria, but depending on the design that you've selected, those methods may not be available or feasible, especially in an observational study. And instead, you have to use statistical methods to address these things. And your protocol should pre-specify how you're going to address the bias and confounding. And it's really important to make sure that you have not selected databases or data sources or specific analyses to favor certain conclusions. The protocol and analysis plan should be established in your protocol prior to conducting any analyses. You need to pre-specify appropriate comparators and statistical methods and ensure masking during development and CVM review of the protocol. So what this means is that you do need to evaluate your data sources to make sure all of the information that you're going to need to analyze is there and those data sources have appropriate fitness, but we're not looking at outcomes or have conducted analyses to kind of know what potential answers would be prior to pre-specifying appropriate comparators and statistical methods. You want to use objective data where possible and make sure that you're conducting a robust data assessment. So this data assessment includes pre-specification of your quality assurance and quality control plans, some validation back to source data, an explanation for how you're going to correct any data and assess for duplicated data, how you're going to assess for data plausibility. This includes like biological plausibility, how you're going to assess for completeness of data. Really important in real-world data sets to assess factors that cause missing data. So missing data are those data that would have been used but were either not observed or not collected or not accessible. It's possible that data could be missing because the data source didn't even have a place to record it. So that wasn't even intended to be collected. It also may be a situation where it was intended to be collected, but the person recording the data didn't record it. So it's just a missing piece of data. Or it was collected and recorded, but somehow it was lost between the source and the final data set. The statistical analysis section of your protocol should include your hypothesis that you're testing, define your statistical models, and provide details of sensitivity analyses that you will perform. Within your model, you need to describe how you're going to adjust for confounding variables and control bias in the analysis of non-randomized data. And it's not in the scope of our discussion today to talk about specific statistical methods, but we do refer you to our guidance. And at the end of this presentation, we provided you with a number of resources to help you understand the different statistical methods that are available. Your methods also need to address missing data, such as with imputation of missing values or utilizing robust methods. And you need to quantify uncertainty using tools such as confidence intervals. Sensitivity analyses provide a way to assess how robust the results of the analysis are to the assumptions that you've made in your model, and how missing data is the impact of missing data and unmeasured confounding. They also evaluate the potential impact if these assumptions are violated. You need to pre-specify all of the sensitivity analysis that you plan to perform, and these will help us to interpret the study results. And finally, the basis of study conclusions should be predetermined in your protocol. And this includes the criteria for concluding that the new animal drug is effective for the proposed indication, how to interpret the comparison between treated and control animals, and or treated animals and a performance goal if that is being used. The criteria also need to include whether statistical significance is required. Typically, this is a p-value of less than 0.05, and how additional pre-planned statistical analyses will be interpreted. The interpretation of these studies requires even more coordination between biostatisticians and clinical reviewers because we're dealing with sensitivity analyses and additional evaluations of model assumptions that just add to the complexity of interpreting these types of studies. So this is going to require extra close communication between biostatistics and clinical reviewers. So we hope that today you gave you a little bit of an understanding of the range of regulatory questions that could be addressed by real-world data and, if analyzed, real-world evidence that comes from that. And we, as has been mentioned in earlier presentations, we really strongly recommend early and close communication with us if this is something that you're interested in pursuing, especially if you're developing a protocol to use real-world data to generate real-world evidence. And we really hope this gives you an appreciation for how to evaluate sources to determine if they have sufficient fitness and they contain the information needed to answer the regulatory question. It's important to make sure you're selecting from all available sources and avoiding selection bias. And also just emphasize again that if you're using real-world data to generate real-world evidence, any protocol needs to be developed in advance and ideally will undergo review and concurrence by CVM. And these protocols need to address confounding factors and biases through both the design and the analysis plan. So the references that we are providing for you today includes a mix of literature, some of which were already provided in our guidance, as well as the link to the real-world evidence page and our guidance document itself. The real-world evidence page includes a number of other guidance documents that have been put out by other FDA centers, some of which have principles that could be applicable to veterinary drug development. Thank you. I'm going to turn it over now to Walt for the Q&A session.
Thanks, Emily. So right now, everybody's coming up who's going to be at the panel for the questions that we received this morning. And as they get situated, there were a few questions that I can answer. And I'm going to pretty much clump them together. And that is, first of all the information that's been presented today, including the slides, copies of the agenda, and anything else associated with the meeting, that will be posted on our website, which I showed earlier today, but it will be posted. They will also be posted in the docket. So I hope that answers your question, but yes, they will be made available. With regard to this morning's questions, just a reminder to folks, we received a lot of questions, which is great, but I don't anticipate that we'll be able to cover all of the questions. And it's also possible that some of the questions require a great deal of additional information that we may not have readily available this time, but we'll take the information as a question that you've received to make sure we address it at some point and consider it at other opportune times to make sure it's made available to the public. And so my order is I'm going to go through the presentations, but I'm going to alternate the topics based on the presentation order. So the first one would deal with the user fees. And I'm going to read this question, and obviously Aila's up at the front to be able to answer your question. And I do remind all the presenters at the table, just press the button on your microphone, turn them on, state your name, and then you can answer the question. So question one. In the workload adjuster slide, it was stated that the percent of direct review hours spent on of regular review hours spent on particular submissions. Is there a possibility to see these numbers? Or the average times of each type of application review?
Okay, so this is Aila Albrecht, and some of that data will be available later this year when CVM publishes the third-party assessment. We're also always seeking ways to enhance transparency to stakeholders. So thank you for asking the question.
All right. Thanks, Aila. The next question probably will be addressed by Courtney, and it's dealing with foreign data. And the question is foreign data also acceptable for reasonable expectation of effectiveness studies to support CNADA?
Uh, Thanks, whoever asked that question, because I love talking about reasonable expectation of effectiveness. So I would say the short answer is foreign data is absolutely great for RXE. What I would recommend if you are considering going down the route of conditional approval, expanded conditional approval, is if you go into animal drugs at FDA and just search for -CA1, you can get all of the FOI summaries from our conditionally approved drugs. And there are a lot of different ways that we've gotten to an answer of reasonable expectation of effectiveness, several of which have used foreign data. Thinking off the top of my head, an example that I mentioned in the presentation, UpCard CA1. Sorry, my examples are going to be cardiac drugs because that's what I work on. But UpCard CA1 used 2 European field studies that to support reasonable expectation of effectiveness. And actually for VetMed CA1, there were US and European sites used in the study. That one it's a very robust study for RXE, so maybe not the best example, but I would just say absolutely. And if you have any questions about it, I'm happy to answer with regards to how that data can be used, because I think there is a lot of opportunity there. Thanks.
Thanks, Courtney. So the next one is related to real world data. I don't know why I have a hard time saying that this morning. So here the question is, for real world data, what if veterinarians use product not in compliance with the labeled use, dose, or frequency? And the second part of the question is, can the data be used to support the label claim? And can it be used to support a label change? And so I'm going to open that up to anybody who was talking. Probably Emily could give you the best thing there on this.
So I interpret the first part of that question to mean that an approved drug is being used in an extra label manner. And that absolutely could be used to help support a supplemental approval. So if, I mean, the veterinary drug has to, or the drug has to be available for use for the veterinarian to be using it in a real world data context. So probably one of the best possible uses, I guess I shouldn't say the word best, but a great opportunity would be drugs that are already approved. Veterinarians are using them extra label, and you can leverage potentially that extra label use to support a supplemental approval for that different species, different dosage regimen, something different about that drug. So, in the second part of the question, sorry.
It's okay. Can the data be used to support the label claim? And then the final question is, can it be used to support a label change?
So if you're demonstrating reasonable expectation of effectiveness or substantial evidence of effectiveness that could support a new claim, that could be for an original approval or for a supplemental approval, there's probably going to be, like for approved drug that's being used extra label, you could get a supplement to that drug. In some cases, there are drugs that are like human drugs that are being used in veterinary medicine that you could get an original approval for, and some other drugs that are just widely used that you could, get an original approval for. But so yes, you can conduct studies using real world data to generate real world evidence to support a label indication.
Thanks, Emily. All right, the next one, I'm going to circle back to Aila, and you can answer it the best way you can. In a situation where an INAD holder has failed to pay the required annual sponsor fees for a number of years, when a new sponsor acquires the INAD from the initial sponsor, will the new sponsor be reasonable for any of the unpaid fees?
Okay, so for that situation, the new sponsor will not be responsible for those unpaid fees, provided that they're not an affiliate of the old sponsor.
Great. That's pretty direct. Okay, the next one is foreign data, and this is open to anybody who feels they can answer this. Um, the examples shared in the presentation, the foreign studies, are all conducted in European countries. And I'm reading these as they were typed. Are any objection or recommendation which foreign countries and or region FDA consider as acceptable country?
So I think the short answer is no. We do see a lot of data that comes from Europe. There are, on the safety side, a lot of big labs that are in Europe that also conduct our studies. I think I also gave one example with effectiveness data that was collected in Canada. You know, the short answer is that there isn't anything specific. It's just sort of a matter of finding a place that will conduct a study to basically to the rigor and what's the word I'm looking for? To the standards that we're looking for, essentially. I think I had an example in there of data being collected in South Africa and Morocco as well. And so it really, if you have questions about it, I would say just come and ask us. But you know, any lab that is using regulatory standards to conduct their studies is certainly one I think that we would consider. If there's more specific questions, I can certainly try to answer that, but I think that's about as much as I can do without more information.
Thanks, Courtney. And again, I'm reading these as I've never seen them before, but I'm doing the best I can at the wording. This one's for Emily. Emily, in your presentation on real world evidence, it says real world evidence may be used for protocol development for a traditional clinical study. Is there any instances or justification that CVM would accept real world evidence in innovative study designs?
So I gave a couple of examples of study designs. There are a lot of others. On the real world evidence page for FDA, there are a series of guidance documents that talk about different types of study designs, that some of which are incorporating real world collection of real world data in a routine care setting, or I'm sorry, in a research setting where you're doing a traditional clinical study, but you're incorporating, you know, some real world data. There's also some examples of the conducting a study in a routine care setting that has some of the elements that are more like a research study. So they're like when I talk about the distinction between real world data and clinical study data, I was kind of hinting at some of those areas where there's blurred designs. But there's absolutely an opportunity for innovative designs that, you know, they're not always just completely real world data. There's a mix potentially of clinical study data, maybe in one treatment group, and real world data in the second treatment group. That was one example we provided. That would definitely be innovative design.
Thanks, Emily. Appreciate it. All right, the next one deals with foreign data. In this section, it was brought up that foreign field studies may be used for substantial evidence of effectiveness, including parasites. What are CVM's expectations for determining that parasites are not different? And then the second part of the question is there any guidance for determining the equivalence?
So I think the short answer is you would need to talk to the respective division about that. There's a lot of different ways we've looked at it. It's hard with parasites and in terms of looking at strains, and I think it was specifically about field study, right, Walt?
Yes.
Yeah, and so I think it's very parasite specific. We have very specific guidelines about heartworm because we know, I think that one is the easiest one to refer to because we have an idea of what differences are between what we see in the United States and what we see in other countries in terms of, resistance patterns and things like that. And so it's very species specific. So I'm not sure I can give much more than that in terms of the field study. The lab study offers us a little bit more opportunity in a foreign site because you can bring bugs from other countries, which is not as easy to do in a field study. But we certainly are open to expanding, particularly in parasites that are not as commonly seen in this country.
Thanks, Courtney. All right, the next one is, this is for anybody on the foreign data side. Can you give some examples of when regulatory authorities' decisions have differed under the simultaneous review / PSA / five-way collaborative review pilot? And there's a second part to it. When this happens, do the applications with different authorities then diverge? Or does the issue have to be resolved with all regulators before the application moves forward? And I can reread that as you need, Brandi.
Thank you, Walt. I think I got it.
Okay.
It mentions all three of the pathways that I talked about today, and I think the divergent decisions, we have the best examples with our simultaneous review with Canada because that follows the full phase review process. With the parallel scientific advice, it is a single point in time, so we're not really in a situation of do we diverge as regulators? We will be. We will give you our advice. You'll know where we agree or disagree, and then you'll continue working with EMA and with CVM. For working with Canada, it is possible we'll make different decisions. I tried to emphasize that this morning, where all of the regulators are going to continue to make their sovereign decisions. In the case of that program, you will continue working with both regulators, as we expect from the very beginning. So if there are additional questions from one regulator, you'll share that same information with the other. And so you do not have to leave the simultaneous review program if there are differences between the two regulators. But please, if you have any concerns and you're in those programs, come and talk to us because we're happy to work with you and have those conversations if issues arise. The opportunities these programs provide is that we can talk to our regulators along the way. And so more often than not, we do align on our decisions. It's just not something we can guarantee. Thank you.
Thanks, Brandi. And then this one may be for you as well. Has there been any discussion with EFSA about applying the PSA approach that is in place with the EMA? And the second part is, this would cover human food safety, toxicology, environmental safety, and medical feeds. Or, this would go.
Thank you, Walt. We are not currently talking to EFSA about setting up any of these simultaneous review, and some of that, the main reason being that we have different scope. All the programs that I talked about are related to animal drug review, and EFSA does not do animal drug review. The products that go to EFSA are not considered drugs in Europe. So all of our European collaboration is with the European Medicines Agency, or EMA, and at this time we are not having conversations with EFSA in a similar manner. Thank you.
Thank you. All right, this next one may end up being with Anna. Where should the sponsor compliance be located in the study file, or the final study report, or the application technical session?
Thank you for that question. So the sponsor compliance statement can be included as a separate file um, included as part of your study files when you submit through eSubmitter. We would just ask that you clarify or provide us with a description where we can find the sponsor compliance statement.
Thank you. And then the final question for this morning. Has the five-way collaborative review pilot been utilized?
Thank you. That one's for me also. And the short answer is no, it hasn't yet. We're very interested in trying it out. We're hopeful that someone will come forward and want to work with us all. We set this up to see if we could find a way for all 5 regulators to work on something together. You'll note the scope is pretty narrow right now. But if you have anything that is close to that you'd be interested in trying, maybe a product is approved in 3 or 4 out of the 5, and you're looking for a new indication, come talk to us. We're very eager to work with our regulatory counterparts. And so if anyone has interest in the program, we're here. Thank you.
Thank you, Brandi. So with that, I'm just going to make a couple of final reminders. Uh, Even though we've covered a lot of questions this morning, please continue to send the questions in because we will keep these and we will refer back to them for future use, as we mentioned this morning as we set up the agenda for this meeting. And so with that, we're going to break for lunch and we will return at 1 o'clock and we'll resume with the afternoon presentations. Thank you a lot. We're going to get started. I want to cover a couple of things because it's entirely possible that we have people attending the afternoon session that weren't present in the morning session online. And I want to make sure that folks actually understand how the QR code and the submission of questions works. So for those of you who were here earlier, bear with me on this. But if you weren't here this morning, the viewable screen that every online participant has contains a picture of the video of the speaker, an image of the slide that, for instance, the folks in the room can see. But there's also a static image of the QR code, and it should be in the bottom right corner of the field of view that you have. What you would have to do is, and this goes for folks in the room as well as online, you just take a picture of the QR code. If it doesn't work on your phone for the QR, you can also type in the URL address that's also available on that slide. It will take you to a predetermined location that has, it actually lists all the presentation topics throughout the day. You simply click on the topic that you have a question on, and you would type in your question in the open field as open text. You hit submit. It will then give you a chance to ask additional questions. There's no limit to the number of questions that can be asked. This morning's presentations, we had a lot of questions, and I expect to continue to get questions from the morning, but it's also we're going to get questions throughout this afternoon, and I encourage you to send them. And again, if you weren't attending the morning session, we did use the questions from last year's educational conference to develop some of the topics that we're addressing at this particular meeting. So I wanted to cover those bases. And so at this point, I think we're going to move forward and begin the presentations. The next section that we're going to talk about talks about high quality submissions. We have Laura Moussa and Jordan DeSilva who will be giving a presentation. And at this time, I'm going to turn it over to Laura.
Hello, everyone. Okay, there we go. Volume is probably good.
I'm Laura Moussa. I'm a chemist in the Division of Biotechnology, where I spent the last 8 years. I joined CVM in 2010, with the first 7 years being in the Division of Manufacturing Technologies. And Jordan and I are going to spend some time on what makes a high quality submission. So for this presentation, we are going to be providing recommendations that can apply to all submission types, with more targeted information on specific submission types presented in case studies. And so we'll be going through submission quality, e-submitter, raw data, good documentation practices, data capture, and final study reports. I do want to note that this presentation does include case studies and examples, but we have fictionalized all names and products and made up the information for presentation purposes. They do not represent actual products. That will be clear when you see them. So why is submission quality important? The submissions, study reports, and data used to support a new animal drug approval must be accurate and credible for CVM to agree that a new animal drug is safe and effective. The quality of a submission, including each of its components, directly impacts the efficiency with which CVM can review the information, make decisions, and provide clear, concise responses to sponsors. And so there are multiple aspects to quality, from study design to submission organization. And here we will be focusing on best practices in organization, using eSubmitter, providing copies of raw data, and good documentation practices. Our review process ends when a reviewer has made a regulatory decision and has written their review to document and justify that decision. We're writing our reviews to satisfy the legal requirement, and we know that future reviewers will need to understand our work for the basis of future decisions. And so for these reasons, it's very important that we can rely on the information in a submission. So that's why submission quality matters to us as reviewers. Why does it matter to you? Because submission quality impacts timelines. Poor quality submissions take longer to review and are more likely to be found incomplete or inadequate. Overall, your submission should be clear and organized, with an overview outlining the purpose of your submission. Reviewers need to be able to follow along with the submission. And the through line in this section is going to be that the reviewer needs to be able to navigate a submission to find information. Poor organization is a hurdle that costs review time. With the key point being that as a reviewer, I am not familiar with the submission when I get it, and I'm especially not as familiar with it as you are. And so here we're giving some examples to help sponsors ensure that their submission is easy for reviewers to navigate. Examples include using formatting to set main points apart by bolding, numbering, or making a separate section of the document, providing a table or an outline to describe the study reports and amendments to help us navigate through your submission, or providing a roadmap that explains any amendments or deviations and explains how they did or did not impact the conduct of the study and the accompanying results. This roadmap is a tool for concise communication and acts as a guide to the submission, allowing you to explain the organization of your submission as well as highlight critical information to help CVM locate all the items easily and identify key data. Here we've provided some best practices. Overall, we recommend that you perform some kind of quality control step on your submission before you submit it. This could include building internal checklists to ensure that parts are not missing, or having someone who is not familiar with the submission read it over prior to submission. This last suggestion here is not implying that you need to have a crack team of PhD scientists reading it over. Just anybody who is familiar with the product but hasn't seen the submission can be really helpful to have fresh eyes on a submission to see what is easy to find and what's not. Overall, including a history of relevant submissions, for example, in the cover letter, including the submission identifiers and dates for our reference, providing document numbers, version numbers, and page numbers, as well as effective dates on all of your documents, such as SOPs and protocols. And this is very key, ensuring version control. Including a complete glossary of acronyms or definitions early in the submission. For a technical section, including SOPs for all critical procedures and ensuring that all referenced appendices are included with the final study report.
And in general, you may request a meeting with us to discuss the organization of your submission prior to submitting it. The information across your submission, including e-submitter, the cover letter, and attachments, as well as across the file, so across submissions and meetings and briefing documents, should be consistent. We recommend that you check for consistency, and if you find any inconsistencies, that you investigate the source and provide an explanation. If there are known inconsistencies, please explain the differences and their impacts. For example, if the drug is proposed for oral administration in e-submitter, but the summaries of pilot work reflect intravenous administration, this is something to address. When there is conflicting information, we cannot assume that one is correct and the other is not, so we will need documented clarification, which causes an increased number of emails, amendments, or voided submission, all of which could have been avoided by careful review of the information. If we have to ask for an amendment, reset the clock, or refuse to review or file a submission, that increases the review time and is usually based because there are inconsistencies that are unexplained or something has been changed. We request that you provide a logical, coherent, well-supported justification to support the purpose of your submission. And this is common to all parts of a submission, including, for example, deviations in a pivotal study. If you've identified concerns or gaps, please explain them, including supporting and non-supporting evidence, and draw a conclusion. You're kind of walking us through this step in your submission. So you have this opportunity to explain any issues that could have affected study conduct or data collection. You can address these gaps by referencing peer-reviewed literature or other high-quality sources of information. When you do so, please include copies of any references in the submission itself. The use of lay articles, for example, articles that are not peer-reviewed, do not provide strong support. So we recommend that you read and understand the relevant CVM guidance when preparing your submission. The key takeaway here is that we're asking you to not provide large data sets without explanation, or to provide many journal articles without a description of how each article supports your submission. This will increase your review time. It is up to you to provide reasons to support for each claim in your submission. When CVM reviews a submission and finds it to be incomplete, we send an incomplete letter with numbered comments. When you respond to an incomplete letter, we recommend that you reproduce these comments, including numbering, when you respond. This helps to ensure the completeness of your response, as well as making it, again, easier to navigate the submission, the example provided here. It is key to address each question. It's not adequate to simply reiterate information that was in the original submission. If data or information is available to support your response, then please include it. And CVM might request an amendment, refuse to review, or incomplete the submission if you do not address each question. If your submission includes references to master files, it's very important to have good communication between yourself and the master file holder. In some cases, it may be advisable to submit your master file references early in the phase review process. You can discuss this approach as well as timing of submissions with the Division of Manufacturing Technologies. Overall, communicate with your master file holders to confirm that their letter of authorization has been submitted, to determine whether the master file has been previously reviewed, and to ensure that all outstanding deficiency comments have been addressed before you make your CMC submission. If this information is missing, it can lead to the referencing submission being found incomplete. Here we've provided some submission quality tips for the environmental impact technical section. Overall, the conditions of use should be accurate and consistent. For investigational use of a drug, if the use conditions are still under development, then provide a conservative estimation on range. And all questions in e-submitter with blue dots should be answered fully and accurately. And that's actually a recommendation that applies to all of our e-submitter templates. If information is missing, CVM uses multiple tools to address it based on the deficiency in the information. For all outcomes, these additional information would be necessary for CVM to complete our review. However, each outcome has a different impact on the timeline and the number of review cycles. On the worst case scenario side, we have a poor submission quality that is to the point that the submission is deemed incomplete on its face, in which case CVM will refuse to review or refuse to file. This can cause very significant delays. If information is missing or inconsistent, again, based on the scope of information, we might just request a minor amendment, which will delay the review process until the information is received to allow review completion. And in this case, if the amendment does not contain sufficient information, then the outcome might still be an incomplete letter. For a more involved lack of information, we might reset the clock. This is what happens when a submission requires a major amendment to be complete. So when that major amendment is received, we reset the review clock from day 0. For products that are enrolled in our veterinary innovation program, CVM might use our stop the clock process, which is an informal process that allows us to request a significant amendment and stop the review clock while the sponsor generates the information. Once the amendment is received, CVM restarts the review clock, and the number of days that are left on that review clock are used to review the submission without exceeding the statutory time frame. If you're interested in more information about this process, please reach out to Ask CVM. And then finally, we have the incomplete letter, which will require additional review cycles to complete our review. And in the next few slides, I will be providing examples of how these tools are used. So as an example of a refuse to review submission, we had a sponsor who had submitted their CMC technical section for the first time, and it was 15 pages long. It did not include any master files. Instead, the cover letter stated that the entirety of the drug substance and drug product CMC information was included in that submission, even though it didn't include any information on manufacturing facilities for either the drug substance or drug product. This is a particularly egregious example of an incomplete on its face situation, but it's also a great illustration about how being aware of CVM's available guidances could have saved a lot of time and additional review cycles because the CMC technical section was refused to review. And in this case, the sponsor would have needed almost a year before they would be ready to resubmit. In terms of the human food safety technical section, we've provided some examples of when we would be sending an incomplete letter. So for residue chemistry studies, if the final study report does not accurately describe the study's activities or conducts, with the examples given here, then the inconsistencies can either require studies to be repeated to achieve a technical section complete letter, or additional review cycles in order to get enough information to provide an acceptable letter. For our example for resetting the clock, we're using the target animal safety section. In this case, we are giving the example of a sponsor submitting multiple laboratory studies to the target animal safety technical section, but they did not provide any data from necropsy of study animals. In this case, a major amendment would be required to include necropsy data. Once that amendment is received, the clock is reset, which adds 180 days to the review time frame. And in the case where the amendment does contain the requested data, the reviewer would be able to make the regulatory decision and complete the review. So although you get the technical section complete letter, the timeline is longer, but avoids an additional review cycle being needed. And again, this is another example, we'll just keep saying this, to please read our available guidances. In this case, GFI number 185 very specifically states that gross and microscopic examination of tissues of animals in all dose groups is recommended for investigational veterinary products containing a new active pharmaceutical ingredient due to the small number of animals used and the general lack of other safety information in the target species. And for our example for stopping the clock, we're using a technical section containing final product specifications for a product that is enrolled in VIP. During our review, the reviewer noted that the test methods had not been validated. So in this case, the review timeline was stopped to allow the sponsor to submit their method validation information. The sponsor had to spend considerable time and resources to quickly create and execute their validation strategy. This is complicated by the inability to find commercial reference standards, but they were able to submit the requested information and allow the clock to restart. And in this case, the review time was extended by the maximum amount of time allowed under stop the clock, which is 180 days, rather than requiring an additional review cycle to be found complete. In this case, the outcome could have been avoided by talking to CVM earlier in the development process to determine the validation requirements for their products and by familiarizing themselves with the available GFIs. Now we're going to shift gears to eSubmitter. Before we dig in, just note that FDA's eSubmitter website has multiple resources and documents that may be helpful to you as you compile your eSubmission. This includes some how-to videos. If you do have very specific questions, you can always reach out to the eSubmitter help desk for specific answers. Overall, we recommend that you follow the appropriate eSubmitter template and the criteria under the eSubmitter user guide. We often notice that the responsible official has not signed the administrative cover sheet, so we ask that you ensure that they do so rather than just having the person who's the submitter sign it. Please ensure that you've used the correct document type. For an example, we sometimes see people trying to submit a G submission, but they're trying to submit it under a GC file rather than their actual file, NADA or INAD. The total submission size in eSubmitter is limited to 10 gigabytes, but we've recently increased the individual file cap to 250 megabytes, and the acceptable file types are listed here. When you are compiling your report and attachments, it's really helpful to read the prompts in eSubmitter. Based on the input for some fields, additional questions or screens may populate, and so if you don't answer the template questions, that can lead to confusion and delays. Reviewers have a fairly short amount of time upon submission receipt to check in submissions and identify any missing information and make amendment requests. So the better organized your submission is, the more smoothly this process will go. We recommend that you include a table of contents for your attachments, listing the file names and including a detailed description of the file contents, including in the table of contents where key data elements are. We recommend that you organize the TOC to illustrate the files necessary to review a specific variable, particularly when that data is spread over multiple files. Attaching hundreds of files can be confusing, so wherever possible, please consolidate and include a README file for all programming and data files. When you are attaching things into eSubmitters, please use descriptive and unique file names, less than 100 characters in length, avoiding spaces and special characters. Here we provide two examples of how people could name their attachments. Under example 1, you see plain English names, where it's very easy to follow 1, 2, 3. That's super helpful. Under example 2, you see that the PDFs have not been consolidated. Overall, every page is its own PDF. And if you notice page 7, there's both a page 7 and a page 7 corrected, which will make your reviewer cry. So please don't do that. Overall, there are some file specifications for PDFs. They should not be encrypted. They should not require additional software or plugins to be read, navigated, text searched, text selected, or printed. And they should not contain JavaScript or any dynamic content. As a best practice, we recommend that you run optical character recognition before attaching files. And we remind you that hyperlinks between individual PDF documents are not currently supported. So if you have absolute links that reference across files, these will not work. PDF files and bookmarks is a topic I never thought I would become passionate about, but I have. PDFs that do not contain bookmarks, especially large PDFs, and typically when we see PDFs, they can be 1,000 pages, 2,000 pages long. They're incredibly difficult to navigate without bookmarks. In the absence of sponsor-generated bookmarks, we reviewers have to go through and create our own, which is a time-consuming process. Overall, we recommend that your PDFs have a table of contents, bookmarks, and include hypertext links for each item in the table of contents. The bookmarks should follow the outline of the document and should correspond to section headers and subsection headers appropriately. They should not be automatically generated because this often leads to functionally useless bookmarks. And I have provided an example in this slide. In example 1, you see the first little bit of Romeo and Juliet, where the scenes are appropriately nested under the acts, and you can see how very easily you could follow through that document. Under example 2, you see randomly generated bookmarks where nothing makes sense. And again, it will be very difficult usually the reviewer just has to start from scratch in that scenario. To aid in making consistent submissions, we recommend that you do not include the same data in multiple locations. For example, if you have a PDF attachment, including your stability data tables, then please don't put those same tables in your main module 3 PDF. This leads to us having to put 2 PDFs next to each other and just scan to see if they all are the same. Do not attach a PDF unless additional support is needed. If a PDF is included, then that information should be consistent with what is provided in the eSubmitter report. Inaccurate information will result in additional review time needed by reviewers and may require additional amendments or communication. So here is a very made-up example of information in the eSubmitter report not matching what the sponsor has included in an attachment. Under the eSubmitter report, you see that this is a product that is supposed to turn dogs green by oral administration. But once you open up the PDF, you would see that it's defining the product as an injection that turns cats pink. It's a wicked bad example, but we have seen similar types of inconsistencies, and you can see why that would be concerning. It also, once we see one consistency, it alerts us that there probably is an increased likelihood of further inconsistencies in the submission. So then we understand that we'll probably be requiring more review time to make sure that there are not. Our eSubmitter CMC technical section templates utilize the question-based review method, which is designed to ensure all necessary information is provided by the sponsor. This is a great example of how the templates are designed to populate with required fields based on what has already been entered. So if you don't answer a question correctly, or you just type in NA, you might not be actually populating the field that you need to. So if you check a box to certify that information is provided in Module 3, you should verify that the information actually exists in that PDF before submitting. And as always, if you get stuck, you can contact, in this case, Division of Manufacturing Technologies for help, rather than creating your own workaround that could inactivate crucial follow-up questions or sections. So CVM uses the Animal Drug Manufacturing System, or ADAMS, information in eSubmitter to do our CGMP status checks and to determine whether pre-approval inspections are needed. Missing or incorrect information can delay an inspection, which can therefore delay the completion or approval of a CMC submission. Some common errors that we see are not providing supply chains for all applicable facilities, providing incorrect rules or qualifiers in supply chains, or providing incorrect FEI or DUNS numbers. Under our website in the Resources for Use section, we have information about entering ADAMS information, but if that doesn't answer your question, again, please contact DMT for help. We also use the information in eSubmitter to identify any relevant master files that need to be reviewed. And if there's a missing or an incorrect reference, that can delay the review of that master file, which leaves less time to address any problems in the master file via amendments. If a reference master file is deficient because the problems can't be resolved in time, then this can cause the submission to be incomplete. We often see incorrect file types, VMF versus DNF, VMF versus DMF, or incorrect file numbers, or just omitting the reference to the master file in resubmissions. Now we are going to spend some time talking about raw data. For new animal drug applications, FDA requires the full reports of investigations which have been conducted to show that a drug is safe and effective for use. We've provided information on raw data in our draft GFI 287, where we define raw data as the first permanent recording of an observation and say that whether it's handwritten or electronic, it should be attributable, original, contemporaneous, and legible. We do expect the submission of copies of critical raw data with final study reports to support the approval of new animal drugs, because we review this to reconstruct a study and to confirm the accuracy of the information in the final study report. For both GCP and GLP studies, we have provided definitions. Overall, the similarities are that any worksheets, records, memoranda, and notes of original and firsthand observations and activities of a study that are necessary for the reconstruction and evaluation of the study falls under raw data. In terms of collecting raw data, we do have information provided for both GLP and GCP, and Jordan will be talking a lot more about data collection. But overall, the raw data should meet the ALCOA guidelines and should be provided in English. CVM uses the data collected for a study to confirm that the study protocol was conducted accordingly and supports the results and conclusion of the study. So your study is being conducted to support approval. Your raw data should make the case for why that study actually provides this support. High quality raw data is the strongest support for study conclusions. We recommend that you only provide copies of raw data that are relevant to the final study report. And the e-submitter templates for raw data under H submissions address expectations for raw data submissions for GLP studies. Submitting copies of raw data allows for easier navigation and sorting of data for efficient review when you provide them as CSV or XML files. It can be very difficult for us to analyze tables of data as PDFs. So we do ask where possible that you please submit data as this type of file. As a clinical pharmacology example, raw data could include individual animal blood concentrations with columns for treatment group, subject, sex, dose, analyte sampling time, and drug concentration. In the case of deviations, we might want to filter by the type of deviation, the site, the animal, or some other variable that helps us evaluate data more efficiently. And at the top of the slide, we've provided some examples of raw data that we would expect to help us support your study. Here are two examples to illustrate the benefit of submitting raw data. Under example 1, no raw data was provided. Instead, the sponsor provided reports of the studies performed, with the reports being high level and not including key details that impacted study outcomes, such as formulation or population. In this case, the reviewers were not able to confirm that the results were accurate without this information, and the submission was found incomplete. In example 2, where raw data was provided, copies of raw data were provided, the sponsor provided reports with summaries, again at a high level. When the reviewer noted an inconsistency, the copies of the raw data allowed them to resolve the inconsistency without the need for an amendment, and the technical section was found complete. Here we've provided a laboratory dose confirmation study example of the importance of consistency. In this case, the protocol and the final study report showed inconsistencies. The protocol listed worm counts on day 10, but the final study report stated that samples were recounted and verified 1 week later without providing any protocol deviation. The final study report also included worm count data from counts on days 15 and 20. The raw data showed that the original worm counts were performed on day 10, but the counts indicated inadequate infection of the control group. And it was not clear whether this was because of an invalid model, and it didn't specify why counts were performed on days that were not listed in the protocol. So here the inconsistencies between the raw data, the protocol, and the FSR left gaps in the understanding for the primary variable data. We had a lack of clarity, and the questions that should have been addressed were not. So this led to two concerns: the introduction of bias because of the recount, and concern that the integrity and the validity of the data was compromised. These concerns resulted in a technical section incomplete letter, and in this case, 2 additional review cycles were needed to meet CVM's acceptance of the study. And now we'll turn it over to Jordan for good documentation practices.
Thank you, Laura. Hello, everyone. I am Jordan DeSilva. I am a quality assurance study reviewer in the Office of New Animal Product Evaluation on the QA branch. And I'll be starting this presentation off with good documentation practices. Okay. So what are good documentation practices, or more commonly known as GDPs? GDPs are a systematic procedure of preparing, reviewing, approving, issuing, recording, storing, and archiving of documents. GDPs describe standards by which documents are created and maintained. So how can you apply GDPs to collecting raw data for studies? GDPs are the guidelines that one follows in recording information in a legible, traceable, and reproducible manner. So when you record raw data, you should consider these 7 questions. Is it attributable? Do you know who made the observation and who is recording it? Is it legible? Can everyone read it? Is it original? Is this the first time it was recorded? Is it contemporaneous? Was it recorded at the time it occurred? Is it accurate? Is it actually what it's supposed to be? Is it complete? Does it need any descriptive units? And is it permanent? Will it stand the test of time? Okay. So why do we need these GDPs? GDPs ensure the reliable, consistent transfer of information. They ensure product quality and safety. It complies with regulatory requirements. It fulfills the basic premise that good science is reproducible, prevents the dishonesty and fraud, and is essential for producing quality results. It provides control of processes and improves performance, and it enables important messages to be communicated clearly and accurately. So ALCOA, so you've probably heard this acronym a couple times today. So what is it? ALCOA is the acronym for the key parts of GDP. They are attributable, legible, contemporaneous, original, and accurate. Because the GLP's and GCP's definition of raw data is similar, if your raw data includes and meets these attributes, they would likely be compliant with both GLPs and GCPs. So here are some best practices and attributes that encompass ALCOA. Each individual that recorded each data point is clearly identified. If someone other than the recorder conducted the observation or observed the data point, then it should be documented on who was the recorder and who was the observer. The person who performed the task should review what was recorded for them by the recorder. Manually recorded data is recorded clearly and legibly in indelible ink. Data is accurately recorded at the time it was performed or observed, including the date and time. And the data makes sense, and any metadata, such as units for values, are included and documented as well. Narrative documentation of study procedures, events, communication, and notes to file should be clear and provide a complete, accurate description of each occurrence and be fully attributable. So good documentation practices as it applies to protocol amendments. Protocol amendments are official changes to the study protocol, and these amendments change the conduct and requirements of the study. The GLPs define how to perform a GLP-compliant protocol amendment. All changes in or revisions of an approved protocol and the reasons therefore shall be documented, signed by the study director, dated, and maintained with the protocol. Making a protocol amendment should also meet the requirements of ALCOA. Signatures on that amendment should meet all the attributes, and the amendment should have all required signatures prior to conducting the changes to the study. Just like protocol amendments, deviation should also meet the attributes of ALCOA. Deviation should be recorded, signed, and dated by the investigator or study director, and include the reason for the deviation, the corrective action, and the assessment of the impact on the study. Explanations should be provided for why each deviation is not expected to impact the result of the study. If protocol deviations are not recorded contemporaneously, is the investigator or study monitor fulfilling their responsibilities? We've seen some studies where all the study deviations were noted after the in-life phase and lumped into one deviation. This does not support that the study was adequate and well controlled. Investigators should promptly review and document the review of data capture forms for completeness to possibly prevent protocol deviations being found late. Also, all protocol and/or guideline deviations should be documented in the final study report. So good documentation practices as it applies to notes to file. The common issue we see is that the notes to file are written at the end of the study, after the study is completed, or after study completion to document protocol requirements without any contemporaneous supporting documentation. A common example we see is in the dosing procedure per the protocol required additional checks that the product was administered. Contemporaneous documentation was not located or documented on the day of protocol administration. Then months later, the notes to file were written saying that the additional check was performed. So if the note to file was written months, days, is that considered contemporaneous? No. Was there any supporting documentation, any supporting contemporaneous documentation from that day and recorded during the data collection provided? No. We do note that it is helpful that compliance with the protocol was addressed. However, the delay in the documentation and lack of supporting documentation adversely impacts the reliability of that note to file. Okay. So we all know mistakes happen. So how can you make a correction that is compliant with good documentation practices? The individual who made the mistake should line out the mistake by putting a single line through the entry, write the corrected information next to that entry, provide the reason for change, then initial and date that correction. All corrections should be clear and legible. Correction of any entry should allow the original entry to be readable. Therefore, if a mistake is made, do not correct it with whiteout or anything else that could hide or obliterate the original entry. Erasing and overwriting are never acceptable. The date of the correction should be the date the correction was made, not the date the error was made. Backdating or postdating of information is not allowed as well. If there is insufficient room to write the correction next to the entry, then the footnote method can be used to document the correction and should provide information on the date of the change and the person making that change. So this slide is an example of how to perform the footnote method. So footnotes are used when there is not enough space or room to complete the correction of an error that was crossed out, or provide explanation comments when correcting errors. And to make multiple corrections, and it can be used to make multiple corrections on the same page of a document if the reason is the same. A footnote should correspond with a unique footnote. However, a footnote can be used multiple times if it is the same person, same type of error, on the same day, and is being corrected at the same time. Facility or study-specific correction codes can also be used as the reasons for correction. However, if the code does not clearly describe the reason for change, then additional explanation should be provided if the code does not clearly describe that reason. For electronic records, an audit trail may serve as the purpose of traceability if the history can be retrieved and viewed as part of the current record. Any changes to the data, forms, and records after it's been signed and dated as reviewed, verified, or signed invalidates the reviewed by signature. The document should be reviewed, verified again, and re-signed for reviewing the document again with those changes. Modifications to verified records should be limited to authorized individuals. Documentation of modification to critical data should include a reason for the change as well. So some best practices include making sure all the study documentation fully complies with the protocol and standard of conduct. All study documentation was appropriately maintained and will be able to maintain its integrity when archived. Unexpected events and deviations are fully described in the study documentation. We know there is no such thing as a perfect study, so making sure to document what actually happened is important. Study documentation should fully support all statements and conclusions in the final study report, and the final study report should reflect the raw data and what actually happened. It shouldn't be the protocol written in past tense. And then additionally, all study documentation should possess the attributes of Alcoa. So here are some common GDP errors we see. Illegible and unclear documentation, the use of scrap paper or non-official forms to collect documentation, failure to maintain original documentation, obliteration, scratch outs, and write-overs, lack of corrections or excessive changes or corrections. The lack of corrections can sometimes raise questions because the data looks too clean. The use of outdated or uncontrolled forms for documentation. Technicians should be using the most current form, either in the SOP or provided in the protocol. Study procedures are not initialed and dated by the person performing the task. Incomplete study records or forms. Incomplete explanation of changes to the data entries and how to correct the data entry was confirmed. So some things to consider. So CVM does not expect perfect studies. A clean study is more of an outlier than a study with deviations. So just FYI. Unless there is supporting documentation, it is a recommended practice to use notes to file to provide further clarification and not to document protocol requirements months later. It is recommended that quality control or QC procedures are in place to ensure data are reviewed in a timely manner to ensure quality and integrity. Non-contemporaneous documentation is unlikely to significantly adversely impact CVM's assessment of study quality and data integrity unless the issue is widespread or the documentation in question is critical to the study and other information is not available to address the documentation gap. Okay, so now I will go through an example of an unforeseen circumstance that affects data collection and the best way to document it. So there was bad weather at a site, and due to the effects of the weather, personnel could not make it to the site the next day. The following day, personnel were on site and identified damage. Fen, fencing, and cattle are missing from the pen. The best way to document this in a GDP compliant way is to note that the protocol states study animals will be confined to pens for the duration of the study. Therefore, a deviation should be written the day the animals are observed as missing. As animals went missing and there will be missing data entries, the deviation would be preferred way to address this. The deviation addresses the missing data, the missing animals, and how the situation was handled and the impact on the study for not having this data, and the animals going missing for multiple days. The deficient way to document this unforeseen circumstance is not having a deviation or any kind of explanation written at the time of the event. There are unexplained missing data entries on the data capture forms for these animals for multiple days, and then the data starts being entered again days later. Without a deviation, explanation, or note to file on the situation, from CVM reviewer perspective, it appears that there is missing data. The protocol was not followed, and then the study personnel or QA unit didn't notice the missing data entries. A note to file or explanation of this situation is better than nothing. So how does good and deficient documentation affect a regulatory decision? Good documentation is when events are explained and connection to data gaps are clear, all relevant information is provided, and the conduct of the study is understood. And therefore, informed regulatory decision can be made. Deficient documentation can lead to large information gaps, connections are not clear, the scope of missing information is not clear as well, and then the quality of study conduct is questioned. Therefore, additional information must be requested before a regulatory decision can be made. Okay. So now that we just went over good documentation practices, let's talk about raw data capture and submitting it to CVM. So for a study, there are two types of ways to record raw data, manually where it is collected on paper with indelible ink, and then electronically using an electronic data capture or EDC system, using an electronic device such as a laptop or tablet. Whether the data was collected manually or electronically, it should possess the attributes of Alcoa. Any changes should be clearly denoted, legible, and attributable to who made the change. CVM should be assured of the veracity and integrity of the data from collection to submission to CVM. When submitting copies of raw data to CVM, it should be clear which data was collected manually and which were collected electronically, as well as if any data was collected manually and later transcribed into the EDC system. And then also we should know the accuracy of those transcriptions. This could be identified in your protocol where you say paper is the backup if the EDC system is unavailable. If this occurs, it should be clear in the data captured on paper if it was transcribed into the EDC system or not. If it is transcribed, then a description of how you ensure the transcription's accuracy should be provided as well. When submitting to CVM, if the files containing copies of raw data have to be manipulated or have been transcribed into the EDC system or an XML file, a description of those QC procedures to ensure the file's accuracy should be included in the submission as well. Okay. So this flow chart on this slide is to show the different ways data collection can occur and how it can be exported and converted to be submitted to CVM for review. And these multiple ways can be used is dependent on how the data was collected and what, if any, electronic systems can do, and then any file formats that the system can export it in. So making sure we know the flow is very helpful. So if you use an EDC system to record raw data, there are some things you should consider. Raw data collected in an EDC system should mimic how raw data are collected on paper. The EDC system should meet 21 CFR Part 11 compliance and should be validated and equipment utilizing the EDC system to document output should be validated and calibrated as well. All individuals recording data using the EDC system should be appropriately trained to use that system and ensure that all data generated adheres to good documentation practices. Or when it does not, it is documented as a deviation. Based on the study design and requirements, an EDC system may not be the best way to collect study data, so each sponsor should evaluate the needs for their study, select appropriate equipment and systems, and validate that equipment for their needs. Okay. So all electronic systems are required to be compliant with 21 CFR Part 11. You should state in the final study report that all EDC systems have been validated for use and meet 21 CFR Part 11 compliance. Compliance must be maintained throughout the data lifecycle from collection to archiving and submission to CVM, and should be considered during your validation process. Be sure that you are thinking of the following data generation, changes to the data, submission to CVM, and archival. For GLP studies, your GLP compliance statements imply compliance with Part 11, and any systems that are not Part 11 compliant must be included as an exception to the GLP compliance statement. Additionally, you should describe the vulnerabilities of the system and how you have ensured that the system is adequate for its use. For clinical studies, you should describe the Part 11 compliance status of all your systems used in your study in the final study report. And just like for GLP studies, you should describe the vulnerabilities of non-compliant systems and how you have ensured that the system is adequate for its use. In your submission, you should address how the data files submitted were created. Were they exported in a submission compliant format, or did they have to be converted? If they had to be converted, how did you ensure the accuracy and integrity of those files during the conversion process? For multi-site field studies, organizing raw data in books or files by site is helpful, particularly when the documents included in these files are consistently organized for each case and at each site. For example, the file for site XYZ is labeled as XYZ raw data file 1 study ABC 123. And then each subsequential file for the site contains the same bookmarks and same documents, the case ID, then the owner consent form, physical exam form, quality of life questionnaire, and other case-specific documents in an organized manner. And then this same organization scheme is applied to all other sites. Consistent and clear organization across the submission is very helpful, and this helps the reviewers easily acclimate to your submission and saves time on locating items. So now that we talked about how to collect data, let's talk about how to submit it to CVM for review. When submitting copies of raw data to CVM, data and key associated metadata should be submitted. If it was collected in an EDC system, it would ideally be exported directly from the source EDC system and provided in XML format. If the raw data cannot be directly exported from the EDC system in XML format, then you should describe the process and controls in place for the transformation or manipulation of the data to the final XML format. If raw data were originally captured on paper and transcribed into the EDC system, then a copy of the paper document should be submitted in PDF format, as that paper was the original observation and documentation. Another item that needs to be submitted with electronically captured raw data is the audit trail. It should also be provided in XML format, and it should be linked to the original data points, and that any data modified includes the operator ID, time and date stamp, and reasons for change should be recorded as well. Additionally, the contents of the audit trail should be described in the README file like all other XML files. So this slide is specific to what should be in your final study report regarding data capture. Your final study report should include the names of the EDC systems used and the data collected by each system's validation status, and it should be clear on which study data were collected manually versus electronically, as well as which study data were collected manually and later transcribed into the EDC system. Additionally, information on the archiving and retention of the electronically captured data should be included in the final study report as well. You should include a README file in your submission. You can refer to guidance for industry number 197 for further information on the README file. It lists and describes each data and program file in the submission. As mentioned before, it should also describe the audit trail file, listings and contents if submitted separately from the data files for studies that include used an EDC system. You should describe the systems used to generate XML files for the submission. You should state its validation status and how data integrity was maintained from collection to submission to CBM, and when information is converted if it applies. You should also describe the controls in place after data was exported to prepare for submission to CBM. A good resource for electronic files and their submission to CBM is guidance for industry number 197, documenting electronic files and statistical analysis programs. Some recommendations from this GFI are to convert raw data files to XML files prior to performing analysis using the converted XML files, and document how the XML files were converted using a SAS program or R code, and include a description on what software was used for the conversion. You check if the programs are executable. Make sure all files are submitted and include macros. Ensure the variables are named in the data files. Only use alphanumeric or underscore characters. Don't include special characters, dashes, and periods. And the first character cannot be a number. If the data in the files need to be manipulated, derived, deleted, duplicated, or modified, then document it in the final study report and note the manipulation in the analysis program. If certain variables were derived, include documentation. For example, a portion of the intestine sample was examined for endoparasite count. The original count of the parasites from that portion should be recorded and submitted in the raw data, and then the factor that the original count should be multiplied by to derive the parasite count for each animal should be documented in the analysis program and in the final study report. So I talked about data capture in EDC systems, and I wanted to highlight some of the top findings we see in studies that use an EDC system. So first is an unexplained time discrepancy that's observed when comparing the audit trail date and timestamps to the protocol and other study documentation. And this is usually due to the fact that the server of the EDC system is located in a different time zone compared to the site. And making sure to include that description in the submission is very helpful to avoid this discrepancy. Second is a lack of a description of the quality control procedures used if data was transcribed into the EDC system. Third is entry errors and discrepancies indicating a lack of training on use of the EDC system. And fourth, user roles do not include descriptions of what information each role can access to maintain masking. Your EDC system may hide forms to certain roles. However, without you telling us, we wouldn't know what forms would be masked to certain personnel. So now let's go into a case study. So two animals from the same house were enrolled in a clinical field study. The animal dosing XML file indicates that animal 12XY received the IVP and animal 13XY did not. The owner communications XML file indicates that animal 12XY did not receive the IVP, but animal 13XY did. We have the dosing record and the communication record are inconsistent, and it cannot be determined what actually happened. This impacts the assessment for inclusion in the safety population. This suggests poor record keeping or oversight by the study investigator and possibly the monitor. And the outcome and what CVM will do is CVM would request an amendment for clarification. In this case, in a clinical field study, one animal was not enrolled according to the enrollment XML file. However, the same animal was recorded as receiving a dose of the IVP, investigational veterinary product, in the dosing XML file. So therefore, the enrollment file said the animal was not part of the study, but then the dosing records said it received a dose. Again, this impacts the assessment for inclusion in the safety population, suggests poor record keeping, or oversight by the study monitor and investigator. And the outcome and what CVM will do is request an amendment for clarification. So our third and last case study shows further inconsistencies. So two animals from the same household were enrolled in a clinical field study, CB111 and CB112. The protocol states the animals in the same household will receive the same treatment. However, per the animal dosing XML file, this indicates that animal CB111 received the control product, where animal CB112 received the IVP. If the dosing data is correct, then this is a protocol deviation, and this would be looked for, and the deviation will be looked for. And just like the previous two cases, this impacts the assessment for inclusion in the safety population, safety and effectiveness population for the study, and suggests poor record keeping or oversight by the study investigator and monitor. The outcome this time would be an amendment request for clarification and possibly exclusion of these cases from the analysis. So all three of these cases resulted in inconsistencies in the data for each study that should have been noticed and investigated prior to submission to CVM. The submission should have contained documentation to explain these discrepancies. If the raw data is incongruous, CVM will need to rely on the review of the raw data to reconstruct what actually happened. If this is not feasible, an amendment request is necessary, and if the circumstances is not explainable, the cases could be excluded from the analysis. Okay, so now that we've talked about collecting raw data and submitting to CVM, so now that we've conducted the study and have all the data, now we can talk about the final study report. So what is the final study report? So the final study report summarizes the conduct and findings of the study from the collected data. The standard of conduct, GLPs and GCPs, define the contents of the final study report. When submitting a study to CVM, the final study report, along with copies of critically important raw data, are submitted as well. What copies of raw data expected to be submitted varies. General guidelines are provided in eSubmitter, CVM policy and procedure documents, guidance for industry 287, raw data for safety and effectiveness studies, and other publicly available resources provided by CVM. And any raw data expectations for specific studies and projects can be discussed with CVM. For both GLP and GCP compliance studies, the contents of the final study report should fully and accurately reflect the study and its compliance with the final assigned protocol and the standard of conduct, and be an accurate representation of the raw data. Clearly state and accurately reflect the standard of conduct used for the study, accurately and completely reflect data generated during the study, be consistent, fully address any issues and unforeseen circumstances that may have impacted the outcome of the study, explain irregularities, significant events, and deviations and any impact on the study, describe the amendments, contain signed and dated contributor reports from each of the individual scientists involved in the study. For GLP studies, the archiving location for study material should also be stated and include the signed QA statement. For GCP studies, the final study report should include the date of monitoring visits. Some best practices for preparing your final study report is to make sure you include key personnel names and study roles and their masking status. Also include key study dates or timelines. For field studies, the number of enrolled animals in safety and effectiveness populations by study site. And then dosing as individual case listings for companion animals. Some common deficiencies we see regarding the final study report are listed on this slide. The top deficiency we see is the final study report does not accurately reflect the raw data. The final study report should reflect what actually happened during the study. Another deficiency is missing or not signed contributor reports. All required contents per the standard of conduct is not included. It does not accurately describe the QC procedures in place for the transfer of data to a contributing scientist. This potentially creates a gap in data integrity and of its life cycle. Deviations are not reported to the study director and impacts on the study not addressed. Analysis validation plans not defined or described in the protocol. EDC systems not clearly defined or listed, and data transcribed into an EDC system not clearly identified in the final study report or the submission. Additional common deficiencies does not describe how, in the final study report, it does not describe how Alcoa was maintained during data collection. The archival of data collected using an EDC system is not reported. The validation and or calibration of equipment is not described. Incomplete or incorrect data or calculations in the final study report data tables. For example, errors in number of animals, the mean, and standard deviation for a critical variable. Inconsistencies across the final study report. The data in tables is inconsistent or disagrees with data in the text, and the terminology used throughout the report is also inconsistent. Cases erroneously included or excluded from the safety and effectiveness population analysis without an explanation, particularly after agreement at an inclusion and exclusion meeting, and lack of identification and explanation for changes. Additional deficiencies in the final study report, lack of identification of a specific case involved in a particular part of the study, poor animal accountability description in the final study report, contains an inaccurate quality assurance statement and does not document the QA unit performing protocol required phase inspections during the study for GLP studies. So then now let's look at a case study for an effectiveness study. The final study report for the pivotal effectiveness study, ABC 123, had numerous discrepancies and gaps in the documentation and gaps in data and study integrity. Statements in the final study report did not accurately reflect the raw data or were incorrect. The names of key study personnel listed in the final study report were inconsistent with the personnel identified in the raw data. Procedures described in the final study report were inconsistent with the raw data, for example, dose preparation, randomization, post-treatment observations, and examination. And then these were not documented as protocol deviations. The results described in the final study report were inconsistent with the raw data. The final study report did not reflect what actually occurred from the raw data. For this one, the CVM reviewer was able to piece together information from the investigator statement, deviations, training records, and initials on raw data, which provided sufficient evidence to understand that key study personnel listed in the final study report was incorrect and did not have to ask for additional information from the sponsor. Sometimes it's evident that the final study report was created by copying and pasting from the protocol. Often this may save the sponsor time, however, it contributes towards longer review times if there is inadequate sponsor quality control and quality assurance. The final study report in the second case did not discuss an animal with a clinically relevant adverse event, although the AE was included in a table as an appendix to the final study report. The animal received a dose of the IVP and later that day experienced the adverse event. The owner removed consent and the animal was removed from the study. The animal was not included in the safety population, therefore the adverse event was not part of the sponsor's safety assessment, and no explanation was provided to support this decision. Why that final study report did not include the discussion of this adverse event was unclear. The outcome from this was CVM determined that the adverse event was possibly associated with the use of the IVP and was included in the Freedom of Information summary and labeling. When it comes to adverse events, there are a couple of requirements we have. You should use consistent terminology, check for over or undercounting the adverse event rate based on the use of overlapping terminology, ensure good quality control practices, and check the final study report multiple times for consistency with the associated tables and raw data. Discuss all adverse events relevant to the known toxicity profile of the IVP, regardless of the animal or incident rate of the adverse event. And ensure you would discuss all serious adverse events. The key takeaway from this case is to let CVM know and be transparent. We may reach different conclusions based on the available information, but if you address the results as they are, it saves us time. So we just completed the what makes a high quality submission section. And this slide is references pulled throughout this presentation, as well as any references that can help you with your submission and any collection of raw data. And then, of course, you're always welcome to reach out to CVM if you have any questions. So now I'll pass it to Walt.
Thanks, Jordan. So in this time, it's a little hot. So at this time, what I'd like to do is we're going to take a break, and then we'll resume the final presentation at 2:45. Gives you the opportunity to stretch your legs, visit with folks, and we'll stop promptly at 2:45. Depending upon how long it takes the conclusion of Parker's presentation, we may take a break so that allow us time to kind of combine the questions that we've been receiving, and we will go from there. So I'm kind of leaving it on a tentative note here, but at the very latest, we'll start at the scheduled time, but we may begin a little earlier for the Q&A session. So at this time, we're going to have Anthony Parker come up here and give a presentation on adaptive study designs. There you go.
All right, thanks, Walt. Can you guys hear me okay? Yes, okay. So my name is Anthony Parker. I am a biostatistics reviewer on Biostatistics Branch 1 in the Division of Statistical and Biological Sciences at the Center for Veterinary Medicine. I will provide some information today on adaptive designs for effectiveness studies. Also to note, I am not the resident expert on adaptive designs. However, I will present this information today as best as possible, and I do invite you to contact Biostatistics at CVM if additional information is needed. So the topics to be presented today are Introduction to Adaptive Study Designs, Regulatory Considerations and Challenges, Decision-Making Processes for Developing and Implementing Adaptive Study Designs, a summary, and an appendix that has a simulation example. So the introduction to adaptive study designs. In addition to the presentation today, Guidance for Industry number 268, which is Adaptive and Other Innovative Designs for Effectiveness Studies of New Animal Drugs, provides detailed information regarding the use of adaptive and innovative designs in animal drug development. And the definition for adaptive design provided in the GFI 268 states, an adaptive design is a clinical effectiveness study design that allows for prospectively planned modifications to one or more aspects of the design based on accumulating data from subjects in the study. It is important to remember that Prospective planning is a key element of implementing a successful and acceptable adaptive design. So successful planning and implementation of an adaptive design may make the study a more efficient study with practical advantages of saving resources like time, money, and materials. Successful planning and implementation of an adaptive design may also reduce the number of animals needed to conduct the study. This could be due to an interim analysis, which results in an early stop for effectiveness or an early stop for lack of effectiveness or futility. An early stop has ethical advantages in that it may reduce the number of animals exposed to the unnecessary risk of an ineffective treatment, which supports the reduction principle for animal use. These practical and ethical advantages may make considering the implementation of an adaptive design a more appropriate option. So at this point, I will provide some information on regulatory considerations and challenges of proposing and implementing an adaptive study design. To begin, any proposed adaptive design or innovative method should be consistent with the regulatory requirements regarding substantial evidence provided in 21 CFR 514.4. The proposals for such design should be well-defined and pre-specified. All details should be adequate, clear, and final before the study begins. A well-defined and pre-specified proposal is to be certain the adaptations are not simply ad hoc changes. And you may ask yourself, what are the concerns with ad hoc changes? Well, ad hoc changes can present issues during and after the completion of a study. For example, ad hoc changes may present opportunities for the introduction of bias. This bias may be difficult to assess regarding its impact on study validity and conclusions. And when determined to be non-ignorable, bias may cause study data to be considered not valid for use as initially intended. Regulatory considerations. Some regulatory elements to consider when planning an adaptive design are: one, you should adequately control the chance of erroneous conclusions. And two, you should minimize the risk of statistical and operational bias. And I think both of these biases has been mentioned in previous sessions. But with that said, even when an adaptive design is not planned, it is always important to control bias, statistical or operational. This is due, as stated earlier, the potential impact of bias introduced into a study. However, in a study with an adaptive design where interim information of the study progress is used to make study conduct decisions, there is an inherent opportunity for study personnel to gain knowledge of accumulating data. Also, under an adaptive design, study personnel may also gain knowledge of the adaptation rules. This knowledge of accumulating data and adaptation rules could influence the behaviors of the sponsors, investigators, and participants, where this could represent operational bias. Therefore, in an adaptive design study, it is essential to control how interim information is shared and who this information is shared with. Another procedure which may aid in the successful implementation of an adaptive design is your communication with CVM during this process. Communication with CVM should begin as early as possible in this process. And this slide lists several communication methods available to you. Selecting a particular communication pathway may depend on several factors which you should carefully identify and prioritize. You should also consider that the format of your information and how the information is submitted to CVM may depend on the type of response you are requesting from CVM. Therefore, early consultation with CVM may be needed for guidance on the most appropriate form of communication. During early consultation, additional pathways of communication other than those listed on this slide may be determined to be more appropriate. Again, GFI 268 provides information intended to enhance the validity and interpretability of confirmatory studies and aid you during the process of planning and adaptive design. Also to note that methods for confirmatory studies may also apply to exploratory studies, where exploratory studies often have fewer restrictions compared to confirmatory studies. So just a few notes on the differences between exploratory and confirmatory studies. Exploratory studies most often are intended to generate new hypotheses. Typically, restrictions are less stringent, and the studies may be smaller and more flexible. And also, inferential statistics may or may not be employed. Whereas with confirmatory studies are intended to test the validity of previously formed hypotheses. Confirmatory studies do consist of inferential statistics, and they require the statistical methods to be pre-specified. So from this point forward, this presentation will focus on providing information on a particular adaptive design, namely an interim analysis. So we will continue by discussing the decision-making processes for developing adaptive study designs with attention on interim analyses. So as previously stated, the proposals for such design should be well-defined and pre-specified. Detailed procedures are essential to protect study integrity. These methods are: the protocol should clearly explain the process used to evaluate interim data and justify why certain decisions will be made. The protocol should also specify how the results will be communicated to the sponsor and clinical investigators to minimize bias and protect masking. Interim results can create challenges in maintaining the integrity of a study. Data accumulated during the study or interim results should be limited to those who are independent of the personnel involved in conducting the study or managing study data. Implementing such procedures may reduce the possibility of introducing bias. For example, one such procedure could be, with a planned sample size re-estimation, you could implement a procedure where an independent statistician conducts the sample size re-estimation in accordance with the pre-specified interim analysis, and then provide limited information to select personnel in the study. So planning. So during your planning stage, an adaptive study design proposal should address how the proposed adaptive study fits your product development plan. It should also address why the adaptive study design is beneficial, the type of adaptations planned, the timing of adaptations, the number of planned adaptations, the operating characteristics of the design, as well as the analytical methods or simulations used to explore those operating characteristics. It is important to note here that to increase your possibility of conducting an appropriate study with an adaptive design, CVM strongly recommends that you obtain protocol concurrence prior to beginning your study. Also in the planning stage, in your protocol, the pre-specified details should include the list of items given here, which include how you plan to control type 1 and type 2 error, how you plan to control bias. The design should also include your statistical analysis plan and should include flexibility procedures to address unforeseen issues. So the group sequential design and sample size re-estimation are two frequently used interim analysis designs. The group sequential design allows for early stopping for effectiveness or futility, where futility is a non-effective determination. And sample size re-estimation adjusts the planned sample size based on accumulated data. Key features of this sequential design is that the group sequential design may have one or more planned interim analysis and pre-specified criteria for stopping the study, where reducing the duration of the study and often reducing the sample size typically provides ethical and efficiency advantages. Some design considerations for the group sequential design includes multiple statistical hypothesis tests for effectiveness. And you should be aware that this is likely to inflate your type 1 error rate. Timing of the interim analysis is important and should also be included in your considerations. In planning your timing, you should consider that a minimum sample size may be needed for generalizability of effectiveness results, inferential value and independent substantiation of evidence, and a reliable evaluation of safety. So moving to sample size re-estimation, a practical definition is sample size re-estimation is an adaptation to modify the sample size based on interim results. This adaptation should also be well-defined and pre-specified. A benefit of sample size re-estimation is that it helps to avoid underpowering a study, where underpowered studies may fail to detect a relevant effect simply due to an inefficient sample size. And this may ultimately result in the inefficient use of resources, including study animals. A sample size re-estimation is primarily conducted under two scenarios. Under the blinded scenario, there could be adjustments based on the total event rate, as in oncology studies. Here, if the total event rate is lower than expected, investigators may increase the total sample size to achieve the target number of events. And generally, this procedure is thought to have limited or no effect on the overall type 1 error rate. In the unblinded case, sample size re-estimation is often used to estimate the treatment effect. In this case, overall type 1 error may be inflated. So considerations for sample size re-estimation include control overall type 1 error, avoid overpowering the study, where an extremely large sample size detects an effect so small that it is no longer clinically relevant. You may address this concern by specifying a clinically relevant treatment effect and adhering to that specification. Another consideration is that you should maintain appropriate study conduct and study integrity. Strategies should be adopted to maintain the scientific validity and integrity of the study, control the introduction of bias. The previous example of a third-party statistician performing an interim analysis and our efforts to minimize bias and protect masking. In addition, again, timing is important. You should weigh the pros and cons of your timing decision. If adaptation is conducted too early, you may be misguided by highly variable interim data and unreliable estimates. If adaptation is conducted too late, there may be a limited window for your adaptations. So this slide provides two essential assumptions, one for group sequential designs and the other for sample size re-estimation designs. Here we note the independent increment property for group designs and the conditional invariance principle for sample size re-estimation designs. Also here, established methods refers to the methods that are clearly explained and highly accepted in literature and are often used in human studies. So in regards to animal studies, typical established statistical methods may only be directly applicable to studies with simple random samples. For example, if the study will use central randomization with no other restrictions such as weight blocks or cohorts, The statistical model will only include fixed effects, and hence, established statistical methods may be directly applied. Otherwise, established methods may not apply. And in regards to central randomization, CVM may accept a justification for central randomization, along with appropriate strategies to minimize variability across sites. Also, the acceptability of central randomization should be evaluated by both clinical and statistical perspectives. Proposals for using central randomization will be evaluated by CVM on a case-by-case basis at the protocol stage. So there are some special features of animal clinical studies which may prohibit the use of established statistical methods. For example, or an example, is that animal clinical studies often include multiple sites where the treatment assignment is randomized by site. As a result, analyses are performed with mixed models, which may include site and site-by-treatment interactions as random effects. Analysis model for animal studies may also include other factors like stage of production, for example, pasture, dry lot, grow yard, etc. So in essence, the use of established statistical methods may not be used due to the special features of animal clinical studies. Therefore, in a situation where an interim analysis is proposed, simulations may be the only method to demonstrate statistical properties of your proposal, one of which is the control of the overall type 1 error rate. If simulations are needed for your adaptive design proposal, the following elements should be addressed in your simulation details. 1, does the submission clearly state the simulation's objectives? 2, does the submission clearly articulate the adaptation algorithms and operating characteristics that will be evaluated? Are all the choices of parameters within the simulation submission justified? And does the simulation explain all assumptions and details of your interim analysis? To continue, it should include, does the submission specify how the interim analysis will be conducted and how it will influence decision making? Are there any hidden assumptions or steps the CVM reviewers may not be able to identify? And is the simulation code legible for CVM reviewers? So what overall goal could your simulation study accomplish? Your simulation results may demonstrate that the proposed design is a good fit for its intended purpose. In addition, sharing all aspects of your simulation study with CVM may allow for a more sufficient assessment of your adaptive design proposal. So moving to Appendix A. Appendix A includes general details for an example simulation study. But first, to summarize, in summary, even with all the challenges, adaptive study designs can provide a flexible and efficient method to assess the effectiveness of animal products. However, adequate procedures are needed to protect study integrity. Also, established statistical methods may not be applicable and simulations may be required. And as always, early communication with CVM is strongly encouraged. So with this appendix gives you some general details for a simulation study should a simulation study be needed. So this simulation example is for a study with a binary endpoint, is randomized by site, and has one planned interim analysis for an unblinded sample size re-estimation. A typical simulation study includes these steps: data generation, interim analysis, criteria to determine when to perform the interim analysis, algorithm of your sample size re-estimation applied in the interim analysis, post-interim analysis decisions, computation of operating characteristics. Across a reasonable range, typical information to be provided with your proposal include the expected success rate for all treatment groups in the study, the expected variance of all nuisance parameters, and the expected values of all covariance. The next 3 slides provide information on elements which should be considered and included in the data step of your simulation study. First one, you should include your statistical model. In this case, it would be for binary data. It would incorporate site and site by treatment variability and use the Logis scale to simulate the data. In your data generation step, you should also include number of iterations per scenario, planned and maximum sample sizes, randomization ratio, number of sites and evaluable cases per site, and a strategy for completing enrollment after your interim analysis. And lastly, you should always simulate realistic enrollment and to account for overrun in that simulation. Criteria for interim analysis timing are: perform the interim analysis when a certain proportion of planned samples have completed the primary endpoint assessment. And also consider the number of sites eligible for your interim analysis. If a sample size re-estimation is planned, elements to include for a sample size re-estimation are the statistical model used at the interim analysis, the test statistic used for your sample size re-estimation calculation, and the method to determine your sample size re-estimation. The next few slides remind you to include planned decision options based on your interim analysis. And some of these possible actions include stopping for futility, continue to plan sample size, or continue to updated sample size. For futility, there are 3 major scenarios: binding futility and non-binding futility. The first one, binding futility. The study should always stop if the futility criteria are met, and the clinical study must follow the same rule of binding futility as in your simulations. In non-binding futility, the futility stopping criteria are simply guidelines that may or may not be followed. Futility stop will not be implemented in the simulation, and the simulation to assure the simulation type 1 error rate represents the largest rejection rate under the null. And the recommendation is always to try to use non-binding futility for broader applicability. Again, these are some post-interim analysis decisions that should be included in your proposal. Stop for futility, continue to plan sample size, continue to update a sample size, and specify the statistical model to be used in your simulations. And here, this slide provides elements typically included when you summarize your simulation study. Specify the method for computing operating characteristics, and always give your key metrics: the alpha level, overall type 1 error rate, and your power estimates. And this slide gives some references used to create the slides given in this presentation. And I think that concludes the presentation. Thank you.
Thank you, Parker. So at this time, we're going to take a 10-minute break just so we can pull all the questions together and prepare them for the actual Q&A session. So stay tuned. We'll be back in 10 minutes. I do appreciate the fact that you've stayed and actually, you know, been able to get up and discuss things, ask questions throughout the day. That's very nice, and that's part of the benefit of having the in-person. Um, One of the benefits I'd like to point out is that we have had a very positive, we've had some very positive feedbacks on the use of the QR code, and it's made things much easier for us in preparation for the Q&A sessions. And so let's go ahead and get ready to begin the Q&A session. And at this time, I'd like to bring Parker, Laura Moussa, and Jordan DeSilva up. They're going to sit at the table. I've got a few questions that we received. I will remind you that we received a lot of questions, but not all of them will be answered at this session because some of them require a different level of expertise than what we're prepared to provide at this particular time. And we're also joined by Michelle Corneli, who's going to be able to field some questions as well. Just as a reminder for everybody at the table, if you have a question and you have the answer, don't forget to press the button on the front of your microphone, move towards the microphone, and speak clearly so everybody can hear you. With that, here we go. This one is for the high quality section, and the question is, and again, I'm reading them as they're typed, and I haven't read them yet, so I'll do the best I can to say it correctly, so bear with me. Here's the question. For development submission packages, are abstracts of literature acceptable in large packages of references rather than full articles? That's the first part of the question. The second part, then it says abstracts are acceptable to OS&C. Laura.
Yep. Laura Moussa, we do want the full article, not just abstracts.
Very good. Anyone else need to chime in on that? Are we good? Excellent. Second question, I'm going to skip down. I'm going to give a question to Parker. Are studies that are conducted to support reasonable expectation of effectiveness subject to the same rigor when it comes to adaptive study designs? For example, things like controlling for type 1 error rare, etc.
Hi, I'm Anthony Parker. So if you are designing a study to be used solely for RXE, then the standards of the study may be relaxed. However, you may want to come talk with CVM regarding your plans with that.
Thank you. All right, the next question. It was mentioned that the data should not be submitted in duplicate. I often find that eSubmitter will request specific data or documents to be submitted that are already included in the compiled main submission PDF. What is the best course of action in this situation?
So we do recommend that you become familiar with the eSubmitter templates prior to compiling PDFs. You can go in and look at all the questions, play with the templates. It's really fun. So take the time to be very familiar with the eSubmitter template before you start compiling. If you have compiled and you find that there are places where there's duplicates, first of all, if it's something that we're going to need to analyze and we have requested it in a specific format, please submit it in that format. We do have guidances available for those types of things. If it is in a compiled PDF and eSubmitter is asking questions about it, you can always reference the file name and the page number rather than duplicating the data.
Thank you, Laura. The next question is, the question on study correspondence between a sponsor and a PI. Did you say that we need only submit emails, etc., if they're relevant to the FSR? And then it follows with a statement. We have previously submitted all correspondence so that you can spot check whether the study was run appropriately.
Jordan DeSilva. So copies of correspondence relevant to the study should be submitted. Not just that's what's in to support the FSR, but all study communication, relevant study communication should be submitted.
Thanks, Jordan. The next one. If an amendment to a protocol is required, would it be acceptable to provide a copy of the protocol with track changes to demonstrate the amendment changes made? In addition, would providing a clean final copy of the protocol with all amendment changes included, and in parentheses, track changes accepted, be beneficial?
Jordan DeSilva. A track change version of the protocol shouldn't be a substitution for implementing protocol amendments. I think a track change version can definitely be discussed with CVM prior to submission if you want to.
Thank you. And as we answer questions, please make sure we speak up loud enough so that we can capture for closed captioning online. The next question. What about the program where FDA is given full access to the EDC database and can review the data with all audit trails intact? Do we have to extract data from the EDC?
Jordan DeSilva. I'll first say thank you for bringing up the pilot or remote access pilot program. So that program was started because there were comments regarding the burden of submitting audit trails from EDC systems. So if you're part of the pilot program, anything that I can see, only thing that could potentially be exported from the EDC system are the stats files in XML format. And that can definitely be discussed if you would like to participate.
Thank you. And now the last question. It was recommended that a sponsor should maintain close communication with the holder of a DMF or a VMF in order to determine that an MF has been previously reviewed and to ensure all outstanding deficiency comments have been addressed before making a CMC submission. What correspondence or notification is provided to a DMF or a VMF holder that their MF has been reviewed and all deficiencies have been addressed?
So we have a policy of reviewing master files when we have a referencing submission in-house. The master file holder will know if they have a deficiency letter that they have not responded to, so that's a very straightforward situation. If they have responded to a deficiency letter and have not received any correspondence, we understand that that might be what this question is getting at. The master file holder can always contact CVM and ask what the status of their master file is and then communicate that to you. We're also working on some other methods of keeping them up to date on the status of the master file. And then finally, once you submit a referencing submission, if you get an approval letter, that's a great time to communicate with the master file holder and let them know that their master file was sufficient to support this referencing submission.
Thank you, Laura. Well, that concludes our Q&A. I'm going to advance the slide, and before you all get up, just want to go over a few final things as a reminder as to what we're going to do from this point forward. Just to let you know, the slides and the videos will be available online, and we're going to place them in the docket. More than likely, the docket will have a link to the video, but we will also provide this information in the CVM meetings website, so you'll be able to find it there. Any additional questions following this meeting need to be submitted directly to the docket. As soon as we finish this meeting, we will shut down the QR code system. But all the questions that we've received have been retained, and they will be put into the docket, so nothing is lost, and we will be able to review them next year and for following years, as some of the subject matter associated with the questions may actually be applicable to future discussions. I want to also point out that for those who are interested, the video recording from last year's meeting is actually is available, and you can actually find that through the CVM meetings page, and the link is provided here. Again, as of now, just a reminder, we're going to have the slides and the finalized agenda submitted. Our plan is to have it by this Friday. Sometimes it depends on our availability to get things posted into the website, but that is our goal. We're in a much more accelerated pace now, and we will do our very best to get them quickly. The video recording takes a little longer because we have to go through an index to make sure that the links that we provide for the YouTube itself actually takes you to the proper dividing point that separates your presentations. So then I'd like to just say thank you. I appreciate your attention. I'd like to say thank you again to my team. They were fabulous. It takes a lot of work, just like Matt said. For everybody who's traveling, be safe, and we will probably have one next year, but I don't have a date yet. So at this time, I'd like to say thank you and have a good afternoon and conclude the meeting.